everyone see my screen? PowerPoint?
Yep.
Okay.
Uh-huh.
Sorry, let me get these windows organized here. What's happened? All right. So today what we're going to talk about is a little bit more regarding hypothesis testing and sort of the fundamentals of hypothesis testing. And what you'll see is from this point on in the course, all the things that we're going to talk about are different types of hypothesis tests. So the very first one that we covered is a one sample z-test. And so everything is going to sort of follow this very similar structure where you're going to think about stating your hypotheses. And so just like we talked about, you know, 2 classes ago, state your hypotheses, setting your criteria, computing a test statistic, and making a decision. Those are all going to be the 4 sort of fundamental steps to hypothesis testing. What's going to change from here on out is what the test statistic is that you're computing, because different experimental designs require different types of statistical tests, basically. The other thing that we're going to add to that piece, part of the core basis of hypothesis testing, is effect size. So we didn't quite get to this in our last class, but essentially after you've done a hypothesis test, another metric that you can compute is if you have a significant finding, how big is that effect that that you have rejected the null, you're saying you have an effect, how big is that effect? So that's sort of the follow-up step to after you've made your decision, reject or fail to reject the null. And then we're going to talk about the different types of errors associated with rejecting and failing to reject the null, and then the assumptions. So all hypothesis tests come along with assumptions, or all parametric tests do. And so within each statistical test from here to the end of the semester that we talk about, we'll talk about the way that you do the test. So computing the test statistic, the way that you can compute the effect size. Different statistical tests have different measures of effect size, and the assumptions associated with that test. So different statistical tests have different statistical assumptions. And so we'll sort of see this pattern as we go throughout the course. But for now, you can just think of these things in terms of z-tests. The type 1 and type 2 error, we'll just talk about that today. We won't really keep coming back to that, but this is a problem with all hypothesis tests. And it doesn't matter the test that you're doing, these are always problems with hypothesis testing that we'll talk about. Then if we have time, we'll move on to some of the other types of test statistics. So we've done a one sample Z. There are other tests called a one sample T test. There's an independent samples T test and a paired samples T test. So there are all different kinds of these statistical tests that we'll eventually cover and make and sort of draw out what situations when you use which test. So as I mentioned, we think about hypothesis testing. You have your 4 steps. State the null, alternative, set your criteria, compute your test statistic, and then make a decision. So if you remember back to our example here, where we had IQ scores, so we have a population mean of 100, standard deviation of 15. If you remember, I took this example straight from lecture 6. So we tested this mean of 103 to see if it was significantly different from the population mean, right? And so do you remember what the formula was for this? So this is a one sample Z test. Do you remember how we did this? So I want to compare my sample mean of 103 to my population mean. So what is the first thing I should do? So I want to to compute this test statistic. So I want to state my hypothesis. What's the hypothesis here?
Null, like the null hypothesis will be sample means is equal to the other means. There's 2 means, right? Population means. Yeah.
Exactly. So null is that they're equal. The alternative is that they're not equal. Then if we want to test our criteria, if this is a 2-tailed test at a alpha of 0.05, what does that put our Z critical value at?
One we put like 0.975 and 0.025.
So those are the proportions. Do you remember what the Z, if we convert those proportions to Z values?
Plus 1.96
yes.
minus 1.96.
Yes, so positive or negative 1.96. So everyone remember we have the 2 tails of the distribution, and then we're going to compute the test statistic. And so this part is where we want to see how different these 2 are. So we subtract 100 minus 103, or 103 minus 100. If it's two-tailed, it doesn't matter which way you do it. And then we have to divide by something. Do you remember what the denominator in all of our test statistics are?
Standard error.
Standard error, right? Because this is the estimate of the standard deviation of our sampling distribution, right? So the sampling distribution has a standard deviation, and we call that standard error. And so we get that by, or we estimate that by taking our population standard deviation and dividing by what?
Square root of n.
Square root of n. So if our n, and our n, this is our square root of our sample size. So square root of 100 is 10. 15 divided by 10 is 1.5. So essentially we end up here with 3, because 103 minus 100 is 3, divided by 1.5, which is 2. Right, so I know I kind of like flew through that, but this is all the things that we did in our z-test in lecture 6. In a second, I'll show you guys this also in R. So 2 is greater than 1.96. So what would your decision be?
Reject the null.
Reject the null. So you would conclude what?
There is a difference between means that is beyond that you obtain by random chance.
Exactly. So there is a difference. So we would conclude that yes, the IQ scores for these students of our sample is significantly higher than the population mean, right? So the next follow-up part to this, so that's our sort of core hypothesis test. The follow-up to this is, if you have a significant result, is to calculate effect size. So the significance of the result just tells you that your probability was low enough to reject the null, right? That you're saying, okay, I think that we had a small enough p-value that it is unlikely that our sample came from this population, right? We're concluding that it's significantly different. But that doesn't tell us how different they are. It just says that they're different. So if we want to know the extent to which they differ, we measure effect size. And so effect size is what tells us what is the magnitude of this difference once you found a significant difference. If you failed to reject the null, you don't have to calculate effect size, which makes sense, because if we fail to reject the null, we conclude there is no significant difference or no significant effect. We don't need to calculate the size of an effect that isn't significant, because it's not statistically there, basically. Does that make sense?
Yes.
All right. So effect size, this can be, so we want to see how big of a difference is between our sample distribution or our sample, which we drew from this sort of hypothetical distribution, right? So we're saying that these two numbers came from different populations or different distributions. Our null here is our population distribution. This H1, think of this as our hypothetical alternative. This is when we're saying that there is a difference between these two populations or these two distributions. The means of these distributions is what we're going to calculate the distance between them. So we want to see how big of an effect or how different are our means from our population mean to our sample mean, presumably coming from this hypothetical different population distribution. So you can think about it in this way, where how far this parameter is shifted, or you can also think of it in terms of the percent of variance that can be accounted for by our explanatory variable. These are just two ways of saying the exact same thing. What is the percent of variance we're accounting for? Because remember, everything that we're doing is really is how much can we account for divided by error. And error is the other way to say how much variance can we not account for. So this is just saying the same thing in a different way. And so for a one sample z-test, one of the most common ways to calculate effect size is called Cohen's d. And we'll also see this for the one sample t-test as well. So Cohen's d essentially just takes your sample mean, subtracts it from your population mean. So no different than what we did earlier, 103 minus 100. And then you divide by the population standard deviation. So we're just trying to see how far apart are our means in standard deviation units. And this is called Cohen's d. And then so because this was developed in like the 1980s or so. So there are sort of standardized metrics to what's considered a small, medium, and large effect. So if your Cohen's d is less than 0.2, it's considered a small effect. Between 0.2 and 0.8, it's considered a medium effect, and greater than 0.8, it's considered a large effect. I will go ahead and tell you in psychology, most of your effects are probably going to be small to medium because we're just limited in the sheer number of population of the sample sizes that we can collect. So whenever your data is noisier, you have a higher standard deviation, your effect can be smaller, and also the effects that we study in psychology tend to be smaller. They tend to be very nuanced differences. So your value for Cohen's d, whenever it's 0, that means there is no differences between the two means. And then this increases as they get larger, which makes sense because we're subtracting the sample mean from the population mean. And then we're just standardizing it into standard deviation units. So yeah, go ahead.
So if you're doing a z-test and you want to get Cohen's d, you're going to divide it by the population standard deviation. But if it's a t-test, you're going to divide it by the sample deviation instead, the n-1 instead of n-based 1. Is that right?
Yeah. Yes and yes. So in a t-test, you will divide it by the sample's standard deviation which we'll discuss in a second. In a t-test, you do not have access to the population standard deviation. That's when you do a t-test. Is if we don't have access to the population standard deviation, we have to do a t-test instead of a z-test. And so the only standard deviation you have access to is the sample standard deviation. But yes, you are absolutely on the right track to already be thinking of these as sort of overarching steps, and then we're just going to apply them different ways. But the overall formula is going to be the same, where you would just divide by the sample standard deviation.
Thank you.
Yeah. Any other questions? It's a very good question. All right, so if we applied this to our example here, where we had a sample mean of 103, population mean of 100, we would say 103 minus 100, our population standard deviation was 15. So our effect size was 0.2, which by our chart here would say, you could say it's on the border between small and medium. I usually err lower, so you can say it's a small effect, which makes sense if you think about this. If the population's average IQ is 100, and our sample was 103, that's only 3 IQ points higher, right? That's not actually that big of a difference. So you can sort of think about it in logical terms as well. Okay, so I'm going to pop into R really quickly and just show you guys how you would do this calculation in R. Maybe I will. Hang on. The window has disappeared for some reason. Right.
Should we open R as well?
Yeah, if you want to go ahead and open the lecture 7 template, you won't have to load anything in for this since we already have these values. So I'm going to start here at the effect size. So this is the exact same example. Remember, we often call our sample mean X bar. So it's denoted by the X with the bar over the top, and our population mean as mu. And our pop standard deviation is 15. This is exactly what we had in the example. So if we could calculate our Z observed and do all of our statistics here and standard error, but we've already done all that. We know that we have a significant result. So I'm just going to go ahead and say reject null. Our Z observed is greater than Z critical. So this is when we compared the 2 to the 1.96. Everyone remember that? And then, so if you do reject the null, then you calculate effect size. So for Cohen's d, we'll just call this d here. We want to take X bar minus mu. Oh, this is S bar, doesn't it? So we're getting the difference between the two means, and then we are converting it into standard deviation units to see how far apart in standard deviation units these are from each other. Here we can see it's 0.2. Does that make sense? Very straightforward. So it's not complicated at all. And most of the time with these effect sizes, they're pretty straightforward. It's just finding the distance. What's the magnitude of your effect? you want to see how different are your two means and divide by some standardizing measure or standard deviation. Usually it's always standard deviation, but it just depends on the test that you're using, whether or not it's population or sample, or we'll talk about pooled standard deviation in a little bit. So any questions about this? Sort of makes sense.
Yeah, not yet so far.
All right. So, all right. So do you guys think that you need another review of one sample Z? If so, we have another example. If not, I'm going to move on to type 1 and type 2 error. So I'm going to sort of get a feel for the class.
Could we review it again?
So here's one more example. If we do a one sample Z, so we know it's a one sample Z because A, it's all you've learned to this point, but also because you have if you have the population mean and the population standard deviation, and you want to compare your sample mean to that, that's always going to be a one sample Z test. So we want to see if there are more red lights run in an hour in the new city this researcher lives in. So we sample 9 intersections. So what is our sample size?
9.
9. And then we record the number of lights run in an hour. We find on average 4 red lights were run across the 9 intersections. So on average, 4 red lights from our sample. So what does our sample mean?
4.
4. And then we know, I've already just included this in here, on the population average is 3.5 per hour, and the population standard deviation is 2. All right, so very first thing, what is state our null hypothesis? So what is our null hypothesis?
It's that it's not, the red lights are not less, oh, sorry, city research.
The same
Red
amount
lights
of
run in an hour is no less than other city.
Yes, yes. So notice here, we said our more lights run per hour. So that is, you're correct. So our null is actually that less than or equal to, and then our alternative is that, yeah, pop mean is more, no, sample mean, sorry. Sample mean is more than pop mean. Or I got these backwards. Because this is a 1 or 2-tailed test.
It's one.
1 because it's more than.
Yes, 1 because it's more than. So we're going to say 1-tailed test. We always assume alpha 0.05 unless told otherwise. So our criteria, how do we find that?
Sorry, what was the question? I wasn't listening.
We want to find our critical. So we want to find our cutoff. So what is our criteria to know when we're going to reject the null and when we're not going to reject the null?
The null is if it's equal to or less than what was the population means 3.5 or equal to or less than 3.5, and null is if it's more than 3.5.
Yes. So now, but if we want to set the criteria for what our critical value is for when we will reject that, it's either a p-value of 0.05 or really it's a p-value of 0.05 because it's one-tailed. If it was two-tailed, it would be 0.025, right? We would split that. Or if we want to find the z critical value, we do q norm.
1 minus 0.95 or what? 1 minus 0.05. Wait, one of the two.
Yeah, I always have to do this also too, because I will inevitably get it wrong. Okay.
1 minus 0.05 is my guess.
Oops, sorry. What happened to my window just minimized here. There we go. Okay. So,
What's up?
so yeah, so we're thinking if our hypothesis
What's up?
is that the sample mean is greater than the population mean, are we expecting a positive or negative z critical? Our sample mean is greater than our population mean, which means which side of the distribution should we be looking on, the left side or the right side?
Right.
Right side.
Right.
Side, yes. So we need to do 1 minus 0.05. So positive 1.64. The other way we could also do this is using Q norm 0.05 and then just doing lower tail. Lower tail equals false, right? This gives us the same number. Do this. So that gives us our Z critical. So that tells us the cutoff of what value we should be looking for. And then the next thing we do is calculate our test statistic, which is our Z observed. So that's the one sample Z test. That's the test statistic itself. So does anyone remember the first thing we need for that? We need to calculate something before we can do Z observed. But what do we need first?
The standard error.
Exactly, the standard error. So we have our standard error here, and that's going to be our standard deviation divided by what? Square root.
The square root of n.
Square root of n, or square root of our sample size, which we already defined up here. Hopefully I ran these. That did not do that. So you can see here our standard error down in the console is 0.6667. And now our Z observed, we're just going to take the difference between our sample mean and our population mean and divide it by our standard error. So say X bar minus mu, remember, is the symbol for population mean, standard error. Now we can look. So our Z observed is 0.75. So do we reject the null or not? So remember, we're going to compare our Z observed to our Z critical.
Fail to reject the null?
Exactly. We fail to reject because 0.75 does not fall in that critical region, right? So the cutoff, it has to be 1.64 or greater, has to be more
Higher, yeah.
than that.
Mhm.
Makes sense. If we wanted to get an actual p-value out of this, what we would do is p norm of our Z observed, right? If we want to find what is the probability of obtaining of our actual value that we obtained, or more extreme, lower dot tail equals false, and this p-value is 0.22. So remember, what p-value do we look for? It has to be what or less to reject the null, to have a significant p-value.
0.05. Oh, wait, actually, it's one tail.
Point-
So
1.4.
It's still 0.05. 0.05, even though that is the standard, and even though two-tailed is the standard, usually what we're saying is we actually are using 0.025 on either end. But because we're actually, this is just a
Yeah, come.
one-tailed.
Use 0.25.
Yep. So because it's a one-tailed.
Okay,
this uses-
and then that, okay. Huh?
Alright.
So you never about, you never go in the city again? Okay.
Your question?
Oh, okay, nice. That's it. Mm.
Could whoever is unmuted, if you could mute. Thank you. Alright. Okay, so does everyone see how we do this? These are two different ways of saying the same thing. So we're here, we know we reject the null because 0.075 is not more extreme than our Z critical. But the other thing we could do is just take the P norm of our Z observed, and we would actually get a P value out, which is most of the statistics that you see reported, where you see like P equals whatever. All right, any other questions about that? Does that sort of make sense? So here, we would not calculate effect size. Why?
Because we don't have an effect.
Exactly, we don't have an effect. We failed to reject the null. There's no effect, or we're claiming there's no effect is our conclusion. We don't know if there is an effect or if there's not effect, but we're concluding based on our p-value and our statistics and our statistical test, that there's not enough evidence to conclude there's an effect. So there's no reason for us to compute an effect size. But if you were going to, you would again just subtract X bar minus mu divided by Popp's standard deviation. Make sense? All right. So any other questions about this? No. If not, I'm going to come back into PowerPoint and we'll go into type 1 and type 2 error. So as I mentioned, we're in that example I just did, we're saying there's no effect because we're concluding there's no effect, right? We don't actually know, and this is one of the #1 flaws with hypothesis testing, but it's unresolvable because this is the reason we do hypothesis testing, is because we're making inferences. So we don't actually know if there is an effect or if there is not an effect, right? And that's why we're doing the statistics to begin with. We're trying to make an inference based on a statistical probability and what is likely based on that probability and what is unlikely based on that probability. But because we never actually get to see the truth and we never actually get to see the whole population and whether or not there is an effect or there is an effect, every time you are doing a statistical test, you're running the risk of making two types of errors depending on what your conclusion is. So you can make either a type 1 or a type 2 error. But the good news is you can never make each of these errors at the same time, but these are all conclusion dependent. So if you reject the null, you're risking a type 1 error. And if you fail to reject the null, you're risking a type 2 error. And that is always the case. There's nothing really you can do to mitigate that other than replication and replication of studies over and over and over again to see if the effect is real, because that's really the only way we can know if there is actually an effect, which almost never happens the way it should, because you don't get papers published for replicating someone else's study, right? So just by design, science isn't really set up for replication, although we rely on hypothesis testing to have these problems. So type 1 error, this is incorrectly rejecting the null. So whenever we're rejecting the null, think about what the null hypothesis is that we're saying the null hypothesis is that there is no effect. So if we reject the null, we're claiming there is an effect. But if you incorrectly reject the null, that means in reality, the null is true and you've concluded that it wasn't true. That's a type 1 error. So this is generally what scientists regard as the worst type of error, although there are instances where a type 2 could be bad. So type 1, you've said there was an effect, but there actually wasn't an effect, was not an effect. So this would be very bad if, let's say, you found a significant p-value, you published a paper about it, someone else followed up on your study, couldn't replicate it, someone else followed up on your study, couldn't replicate it, and this kept happening over and over. That means you probably committed a type 1 error, and that means that paper that you published actually is false. That means that you said, hey, I found that this treatment works, and it doesn't actually work. And you've committed a type 1 error because based on your statistics, you concluded that there was an effect because probabilistically it seemed like there was, but it's all a probability, right? Whenever you say my p is less than 0.05, you're really just saying there's less than a 5% chance that I drew I would draw the same data that I drew or more extreme if the null was true. So it seems unlikely that the null is true given that low of a probability. So you're saying, I don't think that the null is true because there's only a 5% chance that I would get the sample that I'm getting if that were the case, but there's still a 5% chance. So your type 1 error rate is always equal to your alpha, whatever you set alpha at. So your criteria p-value, that's your type 1 error rate. So you can, another way to think of this is as a false positive. And so you can reduce a type 1 error rate by lowering alpha. So let's say instead of saying, okay, we're going to do an alpha of 0.05, let's do an alpha of 0.01. What is my type 1 error rate then?
One in 100?
Yeah, 1 in 100 or 1%. So you're running a 1% chance of committing a type 1 error if your alpha level is 0.01. But let's think about this. If you lower your alpha to 0.01, let's think about it in the terms of a Z statistic. We know at an alpha of 0.05, just one-tailed 0.05, the Z score is, the Z critical is 1.65. What is it at an alpha of 0.01? If anyone has R still open, you can do a Q norm. So instead of 0.05, if you do a Q norm statement on 0.01, what do you get?
0.01, negative 2.32.
Okay, so negative 2.32. So instead of your critical value being 1.65, it's now 2.32, which is going to be more difficult to reject the null.
The first one.
It'll be the second one because
Second
your,
one.
your Z observed has to be that level or greater than, right? So at 1.65, if my Z observed is 1.7, I can reject the null. But if I lower my alpha to 0.01, a 1.7 Z observed no longer is sufficient to reject the null. Now I would fail to reject the null. So a stricter criteria makes it harder to find significance. But that's kind of the point, because you're making it more strict if you want to lower your alpha. So it's a give and take situation. So the downside of that is if you lower your alpha, you might make your criteria so strict that you miss an effect that's actually real. And that's what's called a type 2 error. So if you, let's say you fail to reject the null because your test statistic did not meet the critical value. Let's say it was not more extreme. So like in our example here, or no, not that example, the one we just did, the park red light example where we had a Z observed of 0.75. So we failed to reject the null, but we also are risking committing a type 2 error, because if the null was actually false, we've failed to reject the null, which means we're in essence saying, no, the null is true. We do believe the population mean and the sample mean are come from the same distribution. But in reality, maybe our study was underpowered. So if you remember our stoplight example, our sample size was 9. So if you have a small sample size, you can think of your sample size as a magnifying glass. So if you're looking for an effect in your data, or looking for an effect in an experiment, for example, the more strong your magnifying glass is, or the larger your sample size is, the easier it is to detect that effect. And so this is all goes back related to central limit theorem. Remember, as sample size goes up, standard error goes down, you get a more accurate estimate of your sampling distribution. Is everyone sort of remembering all this? Sort of coming back?
Yes.
So if your sample size is small and you fail to reject the null, then you may have committed a type 1 error or type 2 error, because perhaps there was an effect there, but you just missed it. So a miss is the other way that you can think of a type 2 error. So you can think of a type 1 as a false positive, a type 2 as a miss. So this is, there actually was an effect to be found, but we never get to know if there is an effect or not. But we missed it either because of the sample that we pulled, or because our study was underpowered. Maybe the way we designed the experiment, we didn't have enough data points, or our data was just really noisy. Whatever it is, we committed a type 2 error. And so whenever you lower the alpha, you lower your type 1 error rate, you increase your probability of committing a type 2 error. So as you correct for a type 1 error, making alpha more strict, you're increasing the chance that you're going to miss the effect, which makes sense if you think about alpha. If we say, okay, I'm going to reject the null when p-value is 0.05 or less, but you're concerned about type 1 error rate, so you reduce that to 0.01. So now you say, I'm only going to reject the null when my p-value is 0.01, and you run your test statistic and your p-value comes back at 0.0156. So now, by rounding rules, your p-value is 0.02, technically, if we round it up, right? So now you fail to reject the null, but you could have committed a type 2 error because you failed to reject the null when it was actually really close to your cutoff. right? So it may have actually been significant, but you don't know because you never get to see reality, which is the underlying problem with all of hypothesis testing. But the problem is by nature of why we do hypothesis testing, because think back to why do we do inferential statistics to begin with?
To try to assume the data that we have for the population, because we can never really get the population numbers.
Exactly. We can never get the population numbers, so we never actually get to see if there's an effect. If for every experiment we could sample the whole population, we wouldn't have to estimate things, we wouldn't have to deal with error. And therefore, if we calculated, if we calculated a statistic, or it wouldn't be a statistic then, if we just calculated a mean difference, if those means were different, they would be different, right? But we know there's error, not just from not being able to sample the whole population. There's also error in our measurements, there's error in experimental design, there's error everywhere. But because of all this error, we have to do this inferential statistics. So we don't actually get to see if there is an effect. The only way you can truly see if there is an effect is if you repeat the study over and over and over again, and you keep getting the same result, you keep rejecting the null every single time, you can very, very safely assume that the effect is real. And then you don't have to worry about type 1 error, because type 1 error is only an issue when the null is true. But if there actually is an effect, the null isn't true, right? Because the null is that there is no effect. The problem is that we rarely get to know this is the case. So type 1 error, it's a problem. So are there questions about this? Sort of makes sense. Okay. So I'm going to show some sort of visuals of this so you can sort of see the relationship between type 1 and type 2 error. So I'm gonna maybe make this a little bigger so you guys can see a little bit better. Okay. All right, so can you guys see this distribution here pretty well? Okay, so let's just think about this top scenario here. So let's pretend that we do actually get to somehow know reality, and we get to know whether or not there is an effect or not within a population. So when the null is true, that means there is no effect. So there's no effect between our, let's say, population and our sample mean. So they don't come from different distributions. They're coming from the same distribution. We're setting our alpha right here. And this, it's like 1.96-ish, because that's our standard two-tailed alpha, right? 0.05. And then this is our rejection region right here. This rejection region is the probability of committing your type 1 error. So what I have shaded in blue, that's whatever's in your rejection region, that probability is also the same probability of committing a type 1 error. So this would happen when there is both of your means that you're testing that they come from the same population. They actually do come from the same population. But let's say for whatever reason, your sample mean and test statistic falls in this area and you rejected the null anyway, you're saying they come from two populations, but they actually didn't. That's a type 1 error. So a false alarm of an effect that wasn't actually there. But let's say that you actually do have two different populations that your data came from. Let's say you have the population and then from our community college IQ scores. Let's say that actually is a different population. That's why they have a significantly different IQ. So whenever if your test statistic falls here, you reject the null. It falls in your rejection region. You don't have to worry about a type 1 error because the null is false. You only have to worry about it in this situation when the null is true. But let's say, okay, the null is false. You reject the null. But let's say your test statistic just barely misses your cutoff here and it falls over into this region. You fail to reject the null, but now you've committed a type 2 error because there actually were two different populations, right? But it just fell on the wrong side of your critical line. And that can happen because your study was underpowered. It can happen just because due to sampling error. It could happen because let's say your alpha was too strict. Let's say that you were only testing at 0.01, or we'll talk about in a little bit. Sometimes when you do multiple tests, you have to correct for multiple comparisons. That can sometimes cause type 2 errors because you're correcting and sometimes the correction is too strict and it can make you reject the null or fail to reject the null even when there is an effect. So this area over here, this is the probability of committing a type 2 error. We often refer to this as beta. So this is alpha because we call our alpha 0.05, you set your alpha. 1 minus alpha, this is just a correct decision. This is beta, the probability of committing a type 2 error. And then 1 minus beta is also a correct decision, but we also call this power. So 1 minus beta is your ability to detect an effect when the null is false. So another way to think about this is the ability to detect an effect when there is an effect. So there is an effect somewhere and you found it. That's called power. And power is influenced by your sample size, the error in your data, as well as the actual effect itself. So larger effects, think back to Cohen's d that we just talked about, larger effects are easier to detect. So think of power as sort of your magnifying glass, or your sample size is really your magnifying glass. And imagine if I always my go-to example is because I wear contacts and I'm very clumsy. So I often fumble around and drop them in the mornings when I'm putting them in. Think about if you're trying to find your contact, you dropped it on the floor. If you wear colored contacts, like purple contacts or something, you drop it on your white tile, that's going to be easier to find, right? So that's a bigger effect than if you have, clear contacts or whatever. So you can think of the size as like the salience. So a big effect, it's going to be easier to find. And if you imagine if you had a really big magnifying glass or a large sample size, it's also going to be easier to find. So bigger sample size, bigger effect, you have more power. Things that you can change, sample size. Things you cannot change, your effect size. The effect is what it is, regardless of, even when you calculate the effect size, these are again estimates, right? But whether an effect is there is just, it is there or it isn't there. We don't get to know that. And how big the effect is, that just is what it is. You can't change, let's say, I'm trying to think of a good example. So let's say there is a known difference. I don't know, what is something you guys study? You have a difference where it's like very common knowledge in the field. Oh, let's say like, a lot of like social psych stuff. So, like the bystander effect. You guys heard of that, where we see like people, okay, I'm pretty sure that they've looked at that over and over again. If it's a known effect, then you can measure the effect size. The size of that effect just is. You can't change how big of an effect that's going to have, right? That just is what it is. But you can collect more data points to help you find that effect if you have a small effect, or if you have a large effect and you just want to minimize type 2 error. So think about a situation in which you're studying something, let's say you're studying something on a very like tiny level and it has an effect, or you're pretty sure because you've done all the research, but you think it's going to be a very negligible effect. So you think it's going to be very, very small, but you think there's something really there because of all these theories that you've been reading for your dissertation or thesis or whatever. What is your best bet of finding that effect? You think you're after a very, very small effect, what is the best way that you can maximize your ability to find that effect?
Increase your sample size as much as you can.
Increase your sample size as much as you can. That is the number one thing you can do. So, and sometimes in psychology, or in cognitive neuroscience particularly, a lot of the ways we do that is increasing the trial numbers. So instead of having 1,000 subjects, we might have 30 subjects each perform 1,000 trials, because the number of trials they're performing are also reducing the error, right? Because that's all going into their average for their score. So, and there are ways around it where if you don't have access to thousands of data points, you can find other ways. So like increasing your trial numbers and then using that to increase your sample size. And then the other thing you can do is minimize error in data collection. So this goes all the way back to the very first lecture that we were talking about, error in your design and trying to minimize that and isolate the variable of interest. So the more confounding variables you have, the more error gets into your data. And then the fuzzier that this is going to be, the harder it's going to be to find that effect. So if you're looking for a small effect, you need to very carefully isolate the thing you're looking for and have as large of sample size as possible, because that's your power.
But can't you really deceive yourself a lot with something like that? Like go, okay, I'm going to make this person do 3,000 trials and they're going to be so fatigued that all your stats are going to look right, but you're not really testing what you think you're testing by the end of that. It's all kind of, you know.
Yeah, that is a very good point. So you bring up 2 points actually. There is some statisticians will argue about this, but there is a thing, such I believe, that you can overpower a study, that if an effect is so small that it's not, maybe you can make it be statistically significant, but is it actually significant in terms of, like reality? Is it in reality a significant, like a contributing effect or a contributing finding, right? If it took you 3,000 subjects to find this effect, what does that really mean, right? So, and statisticians will argue about that. So you can get into definitely an overpowered situation, I think. The second thing is, about fatigue. You're absolutely right in that you want to make sure that you're not fatiguing your subjects for 2 reasons that you mentioned. You are not testing what you're testing anymore, but unlike what I think would actually happen, usually what you see with fatigue subjects is you might not get significant findings, because fatigue, just like not carefully isolating your variables, fatigue becomes a confounding variable, adds error to your study or adds error to your data. And now you're less likely to find a significant effect, because the fatigue is not going to be, it might not necessarily be randomly distributed, right? You might have some subjects who can power through and some can't. If they're in 2 different groups, now you've confounded your data. So it's going to add error and it's going to noise up your data a lot.
So also it can be like an error in the trails. If we do a lot of like trails, participants just memorize it, or just like they perform better if they do over and over again. And it's
Exactly.
Yeah.
So what you're describing are practice effects. So if you have them do it over and over and over again, if you have a bunch of different trials and conditions and you don't properly randomize, subjects will learn the order of your conditions, and then they'll start responding based. Brains are lazy, basically, is what we know. Brains will find the most efficient way to do the task at hand. And if there's a pattern, your brain will pick it up even before you consciously know it. Like, you'll implicitly learn patterns, and you'll be able to start, even if you were doing your own experiment, you could start to learn the pattern. And now you're no longer testing the thing that you think you're testing, right? So I used to be in attention research, but this is a really bad problem, because if things aren't properly randomized, if you're trying to test how people are paying attention, but they're just going on autopilot and just, responding to a pattern, you're not testing what you think you're testing. So this all goes back to the very first lecture when we talked about experimental design and how important that very first stage is, because by the time you get to analyzing your data, if that stuff wasn't done right, it's going to be very difficult to interpret things at this stage.
So you basically will have to balance that you don't make your data set too big, but also that you keep it big enough so you can measure the effect.
Yes, essentially. So I wouldn't necessarily say you have to keep it not too big, because especially in neuroscience, cogneuro, psychology, most of the time, even in psychology, data sets that big aren't a big problem unless you're doing like survey research. Like, I'm talking like 5,000, 6,000 respondents to have like an overpowered study. Most of the time in psychology and cogneuro, we're dealing with like 30 to 50 subjects. The more subjects you can get, the better you are, in general, is a good rule to go by. But I do think when you get into the sort of thousands of, particularly you see it in a lot of social psych settings, or a lot of like market research where they're doing like surveys and they're just like getting thousands and thousands and thousands of data points, they do find significant effects. And I'm not saying those aren't real. They probably are real, but they're very small effects, right? Because they had to have 3,000 people to find those effects. But
I guess then that I think for plants though, that might be necessary, because sometimes even if your grow have like 6 or 7 replicates, 3 end up dying, 1 you cannot use, so you end up with very less at the end. Then you don't have like the
Yeah, yeah. So in
Or,
general, we always strive to get as many subjects as possible, but I do think that I think some people will argue that you can't overpower your study, but that's rarely something that neuro people have to worry about. We're usually just clawing for data as much as we can get because it is our experiments are complicated. You know, even if you're dealing with rodents are expensive. You know, we usually don't have an overpower problem. We have an underpowered problem. All right, so you can see type 1 and type 2 error here. Imagine if you were moving this red line. So this red line represents your critical, your cutoff value, right, of whether you reject the null or fail to reject the null. So if you move this red line over here towards the right, that's going to make your type 1 error smaller, right? But what would that do to your type 2 error?
Increase the likelihood of it happening.
Exactly, you're going to increase it. So as you decrease type 1 error, you're increasing type 2. If let's say we're like, okay, we want to have a really high powered study, so let's decrease our type 2 error. So if you think about moving this red line here left to make this area smaller and increasing our power, so we're making power larger, what are we also doing up here by moving this left?
We're making T1, we're making our alpha bigger.
Exactly, which is never a good thing because that's your type 1 error rate. So in general, what you'll find in science is most people very much hate. So type 1 and type 2 error, most people would argue they're not created equal, that type 1 is the worst thing because type 1 errors are what gets you or what publishes false information. And not whenever researchers are committing these errors, these are not ethical problems. These are statistical problems, right? So the researcher is based on their data, they're concluding the correct result. But it just happens that because we always have this probability of committing it, you don't know it until the study's been replicated over and over or failed to replicate is usually when this happens. And then you say, oh, they couldn't replicate that study. That one was probably a type 1 error. So it is rare because again, if our alpha is 0.05, how often do you think this happens?
5% of the time.
Yep, about 5% of the time is what you're going to see. And then it's only noticed of that 5% of the time, it's only noticed when people are trying to replicate and then they can't replicate. And that's when they're like, oh, wait, this was probably a type 1 error. So generally people will regard that type 1 is the worst error. I would also argue that type 2 is not great because there you can imagine cases where, so you can think of a type 2 error, okay, I just didn't find something, I didn't get published, and oh, that was really crappy, but you know, I moved on and did another experiment. But you can imagine a situation in like COVID vaccine trials, for example, when the vaccine was first being developed, how many potential vaccines were maybe thrown out early on because of a type 2 error. They tested it, it didn't work. They failed to reject the null, it didn't work. They didn't test it any further. They're like, nope, that one doesn't work. Move on to the next thing. Nope, that one doesn't work. Move on to the next thing. So if you think about it in sort of a medical clinical testing, a type 2 error can also be bad because you might be throwing out cures for things, right? And not because of the type 2 error, you miss the effect. And when things like in a pandemic need to happen quickly, you just move on to the next thing. You don't have time to sit there and test and test and test over and over again.
Really, isn't power, I hate power. I guess I don't understand it, but it just seems really powerless. It seems like, you know, you could just move that red line way over to the left and go, I'm going to set my criteria at, you know, 0.65. Whoop-de-doo. I'm never going to, you know, I have superpower and you're going to make it's just like, I guess what you were saying about too large, you know, it just seems phony and meaningless. The opposite of meaning.
Increasing power this way is. So this is not a good way to increase power. The appropriate way to increase power is experimental control. So making sure your experiment is designed with minimal error, no confounding variables, and large sample size. So that will increase your power in an appropriate way where you don't have to mess with your alpha. You're just, if you think about increasing your sample size, you're just get into a reasonable amount, obviously. Like if you're increasing your sample size from having 10 subjects to having 50 subjects, that's completely appropriate. And you know, you definitely should do that, like, because that's it's like looking for a contact on the ground with the lights off versus the lights on, right? You could probably find it with the lights off some of the time, but like, it's gonna be way easier if you just turn the lights on. It'll be way easier if you collect 50 subjects. Does that make sense? But yeah, a lot of times power is relatively, I mean, you can change it with the sample size, but you can change it with your alpha, but it's not a good way. That's a reviewer will call you out on that every time. So not a good way to increase power by doing that. And you can't change the effect size. So that is just an unchangeable thing.
Is it possible, like the more you rerun something, if it's not significant, and you want to check it more times, if you run it a bunch of times, will there be one time where it comes out significant based on like probability?
Is it the same data? Is it the exact same data? Or are you collecting the experiment 20 times? So are you recollecting the data over and over again? Or are you just taking the same data and running that stat in R? So I could copy that P norm statement over and over, and it'll give me the same result every time.
Okay, I guess like rerunning the experiment then.
Yeah, so if you rerun the experiment, so you run the experiment, collect the data, run run or whatever your t-test or whatever, and then you collect the data, run the experiment, collect the data, run the t-test, and then you do that 20 times, probabilistically, at least one, if there is no effect. So again, assuming the null is true, assuming there actually is no effect, then at least one of those times you would likely reject the null.
Okay, so and then that would be fake, right?
Exactly. Yeah. So if you, let's say if you just went out in the street and started like collecting data, let's say you had people answer some kind of survey or something, and you randomly assigned these people to groups. You just said, okay, you're in group 1, you're in group 2, you're in group 1, you're in group 2, and you did that. You analyze that data. Because you randomly assign them, you're probably not going to find differences between their answers, right? You didn't manipulate anything. You just randomly said, you go to this group and you go to that group. So in that case, you could safely assume the null was true, and you probably wouldn't find a difference. But if you did that 20 times, 20 separate times, at least one of those times you would likely find that you reject the null and you claim that those two groups are actually different from each other.
So does that ever happen? And like when it's like an ethical concern, like has that ever happened before where you do that just to get a result so you can publish it?
That's a good question. I'm sure probably. So because p-values generally start at 0.05 is what the acceptable, like that's the high end, you would have to do the experiment like 20 times to do it. And at that point, it almost just seems like it would be easier just to plan a better experiment, right? Maybe I could see if someone was being really unethical and had unlimited resources, but that's the thing, is collecting data is expensive. And if you had to do it 20 times, so imagine how much, especially if you're like in rodent research or something, if you had to pay for rodents for 20 experiments, that adds up. It would be far more likely to if people are going to be unethical, they're way more likely just to change the values in an Excel spreadsheet. Like that, if they're going to be unethical, they're going to do it cheaply, right? Like, maybe someone out there is doing it. If you could collect data really fast and cheaply, then maybe that's happening. But all of my experience has been in the neuro side of things, where things are costly, right? So it's expensive. So fMRI subjects are a couple $100 a person, so it's costly to collect data. So I don't know, you'd have to have a lot of resources to do that, but maybe.
Okay, cool, thanks.
Can you kind of do like in reverse? For example, I do my experiment and I got like excellent result. I got an effect. Can I just do more trials just to check if it wasn't like random?
So do more trials, like just keep collecting data after?
No, the same data. Same sample size, samples, yeah, and
so the exact same people, and just
I guess more trails, yeah. Just to check. Yeah.
Yeah, I guess you could do that. So if you did your analysis and you do find that it is significant, you're saying. And then, so most of the time, I guess you could do that just to test, but it's not, because the way that probability works, because we're generalizing to the population, in order to know if the null is true or not, you really need to resample the population, right? Instead of just bringing them, because you need that variability in your error to capture the whole distribution.
Yeah, I see. Yeah.
And then if you're saying the other, I don't think that you're saying this, but the flip side of that is if you've looked at your data, you see that you have a non-significant, like your data isn't significant and you've peeped at it and then you want to collect more data. If you are going to do that, you are supposed to report that in your methods or results that, like, we did this test, it wasn't significant, we thought it was so we think it's underpowered, we're collecting more data. Otherwise, you're getting into like p-hacking territory where, and everyone has different like opinions on this, but it's very much a lot of people, especially of late in psychology, where we've had this sort of replication crisis, that this is more like p-hacking issues, because if you analyze your data, every subject you add, you just keep adding subjects until it's significant, that's unethical, right? Especially if you don't report it. If you say that in the manuscript and you somehow get that published, then I guess you haven't really done anything unethical, because the unethical part is the deceit. So if you're like, hey, I collected data point by data point and just kept doing this until it was significant, then if a reviewer was okay with that and you got it published, then I guess that's fine. But yeah, but if you're peeping at it, you're supposed to be reporting that as well. And it's okay to say, it wasn't significant at 20 subjects, we went back and collected more data. You see that in journals all the time.
So like, what about we do like a set of experiments with like these like plants or so, and then we see that the data is not significant. So I just repeat the experiment with those same set of those plants. So I just repeat my experiment constantly, like 3 times or so. That's fine though, right?
So you do rodent research though, correct?
Yeah, I do plant science.
Plants. Yeah. So that is a little different, because that's a little bit different where you're dealing with a plant rather than sort of human behavior, if that makes sense. So I'm sure that probably is fine, especially I know like in rodent research, they will reuse those rats because you have to from the other ethical standpoint. So in a rodent situation, you have to use the rats. You can't just use a rat for an experiment one time for one thesis and then kill it, sacrifice it, and carry on and order new rats. That's unethical, because that's when animals are used for research, you should be getting the maximum benefit while doing the minimum harm. So that's like one of the ethical standards. So they do that all the time, where you reuse your organisms. So we'll say like plants, rodents, things like that, because that is really so that is a common thing that happens. But whenever you're dealing with human research, where you can just keep collecting more and different humans and you're generalizing to this population, it's better to redo the data, if that makes sense.
So I have an additional question kind of related to that about redoing, but let's say we have 20 data from a human subject, and then we publish a paper, data's published, and later we redo the experiment and we get 30 more data. And with the first data, the null, we couldn't deny the null hypothesis, but the second set of data, you could, right? In that case, would it be considered, let's say it is in the same lab, were we supposed to combine the data and then do it again? Or like, would it be considered more ethical to do it with the combined results of 50N? Or because I can imagine taking 20 and then taking 30 or taking 50 can posit different results. What would be the most ethical way to do it?
So you collected 20 subjects, you looked at the results, nothing was significant, and that was still published, but you published and said we didn't find any results?
Yeah.
So in that situation, when you collect the next set of results, you can't combine that. So there's also things in journals. Whenever you publish or like present at a conference or like you publish your data in a journal, there are copyrights and all these like legal things that you have to sign, and you have to verify that this data is, if you're publishing a research article or research report, like it's usually called like new research or new articles or something like that. Almost all journals that I've ever published in have a checkbox that you have to agree to, that you have to verify that this data has never been published in another journal. So whenever you publish your data in a journal, you technically own the data, but the journal owns everything else mostly including your soul. So like you sign like all the figures you make. I had to get permission from a journal to put in figures in my dissertation that were it was my dissertation that I published before I finished my dissertation. And I still had to go to the journal
Oh my God.
and get permission in writing and put that in an appendix in my dissertation to say that I had appropriate copyright permission because
Oh my God.
I signed away.
Even if it's your data.
my data, my figure, I signed, but mostly it's a figure related thing. But if you're
Okay.
like these paragraphs that you write, so yeah, so once you've published something, but then the follow up to that is what you would do is say, hey, you can reference that paper and say, hey, we didn't find something here, but we did find something significant here. And it's likely, or it's trending, maybe it was, if it was close, it's close, and we think it's because we increased our sample size. And then if you report that you collapse the data together and then write that up in a new report,
Okay.
you still have to get permission from the first journal. So,
Okay, so I think that brings up another question. I think it might be different when we do systemic review or another something like a systemic review when you're taking data from a bunch of different places and presenting it, right? It's gonna likely have a different result than the original numbers previously had.
yes. So
And in that way, we are taking samples from different places and coming up with a hypothesis, either denying or agreeing with the null hypothesis from different time points, but from the same population. So would that be still
that's fine because that's what they call a meta-analysis. And so
Okay.
in meta-analysis, what you can think of as your sample is essentially you're taking aggregate data from all the articles that you
right?
right? And so, and you don't actually have the raw data because you're getting articles from, all over
Right.
the world. And the other thing is that you always say in your method section exactly where you searched, what your search criteria were,
Right.
and how you
Yeah.
found all this and what the cutoffs were. So that is essentially your experiment. And then the data
Right.
points are the aggregate data from all the
Got it. Okay. So you're using each point, each result as a data point as, okay.
Yep.
I
know it's
and then you sell your soul. Got it. Thank you.
yep, and that's the end of science basically.
Okay, cool. Cool. Love it. Thank you.
So any other questions? Oh, great questions. I know they're tangentially related to statistics, but science.
This slide, before we leave that one, on the bottom part, the making alpha more strict, doesn't that decrease type 1 error and increase type 2 error and decrease power?
Let's see, making alpha more strict. So smaller alpha increase, you decrease, you increase power. Wait, no, when alpha is more strict, you decrease power. Good call on that. Yeah.
And you decrease type 1 error too, right? And you increase type 2 error.
Yes, because you're more likely to miss an effect here. So you increase type 2, less likely to commit type 1. Yes.
Thank you.
Thank you for that. All right. So here's basically your chart. So you can increase type 1, or if you make alpha larger, here are the effects it's going to have. You increase type 1, decrease type 2. It increases power, but that's not a good way to do that. Should probably put like an asterisk or something by that, because it's not the great way to do that. And you're more likely to detect an effect, but that effect is more likely to be not real. So because you're more likely to commit a type 1 error. If you make alpha more strict, you increase your, no, really, I've really messed this up. Sorry, guys. Here we go. I have to re-upload this PowerPoint. You increase, no, you decrease. No.
And that's it.
Yeah, wait, you make alpha more strict. You have a smaller
You
alpha.
increase type 2.
A smaller alpha, yeah. Smaller alpha, you increase type 2. There we go. So if it makes you feel any better, that's how complicated that. The best way I keep it straight is down here. I'm going to show you guys a chart. Here we go. This basically shows when you have reality, what your decision is. So this is the best way to think of it. In reality, which again, remember, we don't really get to see what reality is. We only know through replication if the null is true or not. We can only presume. If the null is true and you reject the null, it's a type 1. If the null is true and you fail to reject, you've just made a correct decision. If the null is false, AKA there is actually an effect to be found, and you reject the null, you've made the correct decision. That's also known as power, your ability to detect an effect when an effect is there. If you reject the null, or if you fail to reject the null when there is an effect, you've made a type 2 error, or you've missed the effect. Make sense? All right. Okay, so some examples really quickly here. So researcher found a random sample of N of 16 of college professors commute with a mean of 15.5. Oh, so I guess we need to test this in our, we can test this in our R error. So maybe I will, sorry, let me look at what time it is. See if this is a good time to take a break. Yeah, so yeah, let's go through this really quickly and then we'll take a break before we get into assumptions. So let's test this in R. So if you want to open R back up, let's see. My R window keeps minimizing here. All right, can you guys see my R Studio? Okay, type 1 and 2 errors. All right, so if you want to follow along, you are welcome to. So we have a sample size of 16 and her sample mean is 15.5. So we have an N of 16 and X bar of 15.5. And population mean is 20 minutes and standard deviation is 10. So this is exactly what we did earlier, right? 20. So what is the first thing that we need to do? State your hypotheses. So her hypothesis is that it is the commute of college professors is significantly less than the population. So what is our hypothesis, or what is our null hypothesis here?
It's equal to or more than the population.
So sample is equal to or more than pop. And then our alternative is that the sample is less than pop. These are means, right? And number 2, we're going to set our criteria. So we're going to test at alpha of 0.05. So 0.05 at one-tailed. What is that Z critical?
Plus 1.96
For one-tailed, that's the two-tailed, and you're close.
Oh,
1.64.
1.64.
Yep, 1.64. So the other way we can note this is using this P norm, Q norm, my bad. Yep, 1.64.
We are only doing a one-tailed test here.
Just here, yeah, just based on the PowerPoint slide, because the hypothesis is that they're significantly less. Usually we do two-tailed, but if it says to do a one-tailed, then we'll do that. Or if their hypothesis is directional like that. So we're going to calculate our test statistic. So how do we do that? This is a one sample Z test. So we need our Z observed, which is what? So we know we need to
Don't we need to find the error first?
we do. We need to get standard error, which is
And the reason why you're doing Z observe is that ZOBS is just
yeah, that's what I
just
meant. Yeah.
differentiating it, right? Yeah.
So what is our sample size here? We need the population standard deviation divided by square root of what?
16.
16, or N, as I've put it up there. So standard error. And now our Z test statistic is going to be our X bar minus mu divided by standard error. So our Z observed is negative 1.8. Oh, we're looking at less than, so our Z critical is negative 1.64. So what is our decision?
It's more extreme, so we can reject.
So we reject the null. So now let's do this one more time, but let's do it at an alpha of 0.01. So I'm just going to copy this down here, and I'm going to say alpha 0.01. What is the only thing I need to change? I'm doing the exact same statistical test, except I've changed the criteria. So where do we need to change? Which step is that at?
Q norm?
Yep, Q norms. Let's change this to 0.01. Let's change this to 0.01. So our Q norm is now negative 2.32. And our Z observed, again, that hasn't changed, is negative 1.8. So now what is our decision?
We can't reject.
Exactly. So now we fail to reject. So this is what I'm saying whenever I said making your alpha more strict makes it harder to reject the null, right? So we lowered alpha to 0.01. That makes your Z critical larger, which means in the exact same statistical test, this changed this from a reject the null to a fail to reject the null. And nothing changed about the data. That's a researcher metric, right? So yes, we've lowered our type 1 error rate, but if we make it too strict, we might be missing an effect that's real. Does that make sense? And this is the same thing with what we talked about, one-tailed versus two-tailed. So if these were two-tailed tests, that makes it even more difficult to reject the null, right? Because a two-tailed test splits your alpha. So if this was a 0.01 and it was a two-tailed test, really this Q norm would be 0.05, right? Because we would divide it by 2. And this would be our critical value. Does that make sense?
For the first part, how much did you get the Z observed?
Where did I get the Z observed?
Yeah, no, what was your value Z observed?
Negative 1.8.
I'm not getting that. Let me check.
Do you have your parentheses in the correct spot? This is almost always the first error. It's always my error also, is because if you just do X bar minus mu divided by standard error, R, because of order of operations, will divide mu by standard error first and then try to see.
Yeah, I think that's what I'm doing.
So I can pop this into the chat.
Oh, I can, yeah, I think I figured it out. Let me try again.
All right. Sorry, I just now saw the chat, guys. I posted that in the chat. Sorry, I see I've missed quite a bit. Okay, so are there any questions about that? Does that sort of make sense? How your alpha rate can change your decision in general, but also it's changing your type 1 and type 2 error rates. All right, so it's right at 6:30. So let's take like a 5-minute break. So we'll come back at like 6:35 and resume the recording and let's go back to our PowerPoint. All right. So essentially we talked about our type 1 and type 2 errors. So when you do hypothesis testing, regardless of the test that you're doing, this is always going to be an issue. Always something you sort of have to think about in the back of your mind whenever you're, and this will come up later whenever we talk about more complicated tests and we have to start doing multiple tests, then we run into a problem called multiple comparisons where every time we're testing, it's increasing our type 1 error rate. But we'll get to that later in the semester. So here are some examples that if you guys want to go through yourself, essentially testing whether or not they're type 1, type 2. Always think of type 1 as a false alarm, type 2 as a miss situation. So then the last thing that we talked about with all, or that we'll talk about with, that are going to exist with all of your hypothesis testing are assumptions. So for one sample Z and also for one sample T test, there are 4 assumptions. The number one assumption of all the tests really that we'll talk about in this semester, because we are dealing with parametric statistics and parametric tests, is normality. So that's always going to be the very first thing that you check for is, are your data normally distributed, or are at least your errors normally distributed? The other thing is random sampling. So we talked about this at the very beginning of class. You always want to make sure that your data is sampled randomly because of the bias that can occur, right? So a small sample size also has this problem, but if you are going to have a small sample size, then make sure it's truly randomly sampled. So this is something that you can't change after your data is collected. So this is a during data collection situation. Then you have independence. So this is an assumption that you'll see pop up at various tests throughout the semester. So independence is the assumption that the probability of each outcome measured in the study are independent. So the sample, so for example, for one sample Z test, if I collected data from one sample, from a sample of students, let's say, no one student was measured twice, right? So all the measures I got, so let's say I'm calculating students' GPAs and I want to measure them, or IQ or something, and I want to compare it to the national average. I didn't accidentally sample the same student twice, and their IQ isn't in the data twice. So I have 2 measures that are actually the same person. That would violate independence. So for one sample Z and one sample T, independence is an assumption that you have to have. And then finally, the data are measured at least at the interval level. Sometimes you'll see data on like a Likert scale type thing that aren't technically interval or more categorical treated as interval, but for most of the things that you're measuring in Cogneuro, where you have reaction times, accuracy, response rates, things like that, then you really don't have to worry. Your data is going to be at an interval level. This goes back to, remember we talked about ordinal, nominal, interval, all the ratio, all the different types of data. All right, so all parametric analyses have some assumptions. If you violate these assumptions, if all your assumptions are violated, then it doesn't matter what your statistical results are, because you can't draw conclusions if all of these statistics rely on these assumptions to be valid, right? So we can make these inferences assuming these things are true. If you violate all 4 of these things, it doesn't matter if you have a significant p-value, it's not a valid, that you shouldn't have been using that test because you violated all the assumptions of it. So if you have a data set that you're planning on doing one analysis with and you start violating assumptions, sometimes there are remedial measures you can do. But for example, sometimes there aren't. Like, let's say you're going to analyze your data and then you realize, things weren't sampled randomly. Let's say you're testing the difference between, let's say the difference between students from Queens versus students from Brooklyn, but you accidentally tested all the Queens students in the morning and all the Brooklyn students in the evening. You didn't randomly sample, you didn't counterbalance things. You can't fix that, right? Like, it's a confounded study. So you might be able to add in a covariate, I guess, technically, to account for that. But like if you weren't, or let's say you randomly sampled and you only sampled from one neighborhood in Brooklyn, and then you randomly sampled from a bunch of neighborhoods in Queens, that's not random distribution. So if you violate this, then that's problematic. Things like normality, you can sometimes get away with depending on how skewed it is. There are transformations you can do to your data to help with this. So it depends on the assumption that you violate and how egregiously it's violated as to whether or not you have to move into non-parametric statistics. And so we won't really get into many of these in this class, but a lot of non-parametric statistics are things like permutation testing or chi-square. Sometimes you'll see is referred to as non-parametric. So there are a bunch of different statistical tests that don't rely on these types of assumptions, that sometimes if you violate these, then you have to jump into that category. But for all purposes, we're going to pretend like our data is nice. And for the most part, it usually is. A lot of this stuff is determined at the experimental design level. So as long as your experiment is designed, your independence, random sampling, that's something that you can control at the beginning. All right, so your parametric test statistics, they assume random sampling, independent observations, normal distribution, and one last thing that I didn't mention is homogeneity of variance. And so homogeneity of variance refers to how the variance is distributed within group or across groups. So we haven't really talked about different groups yet because this isn't an issue for one sample z-test, right? Because you just have a sample that you're comparing to a population. But eventually we're going to get to where we talk about comparing two groups. So let's say you wanted to compare Queens students versus Brooklyn students, right? These are two groups. We're no longer looking at the population. We're taking two samples and comparing those. Or let's say you have a treatment group, a drug A group, a drug B group, and a placebo group. And you want to give, drug A, drug B, and a placebo and measure performance in 3 different groups. Homogeneity of variance is the assumption that the variance within each of those groups is equally distributed. So you have similar variances within your groups. So if you can imagine in the drug example, you had drug A, drug B, and a placebo group, and your drug A and drug B group both have very, very small standard deviations. So everyone's real tightly clustered around in the distribution, and the placebo group is all over the place. Now we have a problem because the variances differ across your groups. So homogeneity of variance, something that you have to consider when you have multi-group data. You'll never have to consider this with a one sample z-test though, because there are no groups, right? You just have that's what the one sample stands for. You just have the one. So there's nothing for it to be homogeneous about. All right. So random sampling, all statistical tests assume that your data are sampled randomly in order to capture the population. We talked about this at length, first or second lecture, about how important random sampling is and that how selection bias can, if you're only sampling from one end of the distribution. So let's say that you were, back to our IQ study, let's say that you were wanting to test the IQs at a community college versus the general population. And then you went to sample that college and you went to give out IQ tests, but instead of just, let's say, standing in, the common area and just giving people IQ tests, you went to all the honors classes and said, okay, I'm going to give out my IQ tests. Would your conclusions then be valid that students at that community college have a higher IQ than the general population?
No,
Why not?
because you went to a select group within that general population.
Exactly. You have selection bias. You didn't randomly sample that population, the community college population. You went to all the honors classes. So you went to where all of the likely higher IQ students were and took their IQs, right? So that's a selection bias, and that's a very intentional selection bias, right? But this can also happen occasionally if you're just not thinking about the way that you're selecting your population or your sample size. So a lot of times time of day can be something that people won't think about if you're only selecting certain groups in the morning versus the afternoon. And some people are morning people and some people aren't. So things like that. So random sampling, this is very important one. And there's no way to correct this. If you went to that honors class, collected all that data, there's no way that we can fix that. Now your conclusions just have to be that honors class students at this community college have a higher population than the general population, have a higher IQ than the general population, right? You can no longer generalize to the community college because of the way it was sampled. Independence of observations. So again, this is at the design stage. So all statistical tests assume this. With all the statistical tests that we'll be talking about for right now, except for one exception, assume that your observations are independent. So again, like I was saying, I didn't record the same student's IQ two times or 3 times. I made sure I was sampling without replacement. But one common case of a non-independent observation is in repeated measures design. So sometimes it's really helpful if you have a situation where, let's say I measured someone's performance on a test. I gave them some sort of intervention. So let's say I gave them like a study guide or something, and then I measured their performance again. Then I want to compare those two things, right? So you also see this a lot of times in drug studies where you have a starting performance. Let's say you have starting anxiety, you get a treatment for anxiety, and then you measure anxiety again. So you have a pre-test, post-test design. That's a repeated measure. Those before the test and after the test, that's measured from the same person. Those aren't independent. And that's okay, because that requires a different type of statistical test. So that statistical test does not have independence as one of the assumptions for it. But all the other ones that we talk about in general have independence of observations. So measuring test students over time, as we just talked about. So if you measure them multiple times, that's a violation of independence. And again, this is, you only have this from good experimental design. So if you realize that this has happened, then let's say that you've realized that you didn't, I don't know, properly screen your data and the same person was tested twice, which is again very rare in our neuro research because we have such low sample sizes. Then you have to do something with that person's data. You can either just throw them out completely, maybe you can average their results together from both of their performance, something. They can't be in the data twice if everything else is supposed to be independent. All right, so we have random sampling, independence of observations, normal distribution. So we talked a lot about the standard normal distribution and distributions in general. So a normal distribution shouldn't be a novel concept at this point. But so whenever you get your data, the very first thing you want to look at is look at how your data are distributed. This is not only a good practice for looking at the normality assumption, but it's also good in telling you if there were mistakes in coding the data or if there are any crazy outliers or anything crazy going on in your data. So you always want to sort of plot your distributions, just look at your raw data and see. So let's say you had a person that, let's say you had them do a cognitive test and they were doing a bunch of trials and you took the average of their trials, their average reaction time or something of their trials. And you plot the distribution and you see that one person was responding every 100 milliseconds, which we know is not really possible, right? So people can't do the task and give a response in 100 milliseconds. That's too fast. So that means they were just sitting there clicking the button nonstop, not really doing your task. Plotting the distribution of your data will make that very apparent because their data will be way out on the tail relative to the rest of your data. So always a good practice to plot the residual or plot your distribution of your data, but then also plotting the residuals from the analysis, which we'll get to once we start talking about t-tests and ANOVAs in the next lecture. So it's not only that if your data is non-normally distributed, but the residuals or the error terms are normally distributed, then that's fine. So this is most easily looked at with something that we call a QQ plot or a quantile plot. And in just a second, I'll go into R and I'll show you an example of this. There are some tests that require the data itself are normally distributed, like t-tests, but for others, just the residuals will be. And that's more once we get into like ANOVA and regression-based things. All right, so we have independence, random sampling. What was the other one? Independence, random sampling, normal distribution. Oh, homogeneity of variance. There's more about normal distributions. Sorry, I forgot I had all this in here. Yes, so let me go right back to normal distributions really quickly. So the residuals from the analysis are called the errors. So this is the difference between the observation itself and the value predicted by the model. In all of the tests that we're talking about until we get to regression, the model is the mean. It's a very simple model. It's just the mean. It's the mean divided by the error or standard error, right? So it's like the simplest form of the model. So the residuals are called the errors. So what our observation is, the difference between that and the actual value and what you predict it to be, that's the error. If those errors are normally distributed around the mean, because this is our model, then you can say that your data is normally distributed and it meets those assumptions. So we always look at the distribution of the residuals, not the raw data, because often you have non-normal distributed raw data. Reaction time is actually a great example of this. Reaction time tends to be skewed. Does anyone know which way it's skewed? you think about reaction time in general.
Slow,
Sorry, what?
slow, like later than the,
So think about a leftward versus rightward skew. Which way do you think it's skewed? If you're plotting the distribution out.
so left skew.
Leftward.
Wouldn't it be skewed to the right?
Yes.
Because like
so-
that's more time.
Exactly. So yes. So rightward skewed reaction time is almost always rightward skewed. And it is, but you are right that it's slow, but think about slower reaction times or larger reaction times, right? And larger reaction times are going to pull out in the right side of the tail. And this is because people can only respond so fast, right? Like the fast, even in just a pure detection paradigm, you might have like what, 200 millisecond responses or something. They can't respond a negative amount of time, and they typically can't respond even less than 200 milliseconds, usually 300 a lot of times. And then, but sometimes people can zone out and miss a trial or not pay attention or take a lot of time to respond. And so if you have the earliest response at 200 milliseconds and some of your responses all the way at, 1,000 or 1,200 milliseconds, if they're taking a second and a half to respond, your data is going to be extremely rightwardly skewed. So a lot of times with data like this, it depends on what you're doing, but a lot of times if you have multiple trials within a person, you take their median reaction time and then you average those values together. So remember how we talked about the median will still be the middle, even in rightwardly skewed data. But even if you don't do that, all of this is to say, this is why we look at the residuals, not the actual raw data, because there are plenty of constructs in psychology, or in Cogneuro, like reaction time, that are not normally distributed. But if the errors or residuals are normally distributed, then that's okay. And we'll show an example of this in R in just a second. Actually, here's a good example right here I have. So if you have your mean of 10, one observation is 12, then that residual is 2. And then another observation is 7, then the residual for that is -3. Does that make sense? So within your data, it's how far away each data point is from the mean. Those are your residuals. So as long as they're normally distributed around the mean, then that's fine. All right, so why does this matter? Normality is probably one of the most important. Obviously, independence and random sampling, all those are very important, but those are things that are very much within your control. You can't necessarily control, you really can't control how your data is distributed. The data is the data. All the experimental design things you can control at the beginning of the experiment. Normality is the most important thing that you really can't control, but it matters because of things like central limit theorem. So remember, as your sample sizes get larger, your distribution approaches normality, and the sampling distribution also will approach normality because of your increasing sample size. But in small samples, you have a problem where your sampling distribution is not necessarily normal. And think back to what we're doing here, all the way back to making our decision. Are we rejecting the null or failing to reject the null, right? This is all based on a sampling distribution. Remember, these are these hypothetical distributions. So we know where our critical lines and cutoffs fall on these sampling distributions because we assume they're normal. If you have a small sample size and then your sampling distribution is not going to approach normality, now none of this is going to hold. So now your 1.96 doesn't actually reflect the proportion in the tail that you think it does if your sampling distribution is non-normal, which it would be if you have a small sample size, or it could be. Does that make sense? All right. So second reason, so you have central limit theorem, it's first reason. This is important because of central limit theorem and what we're basing our sample size of. Generally, people like to say the rule of n of greater than or equal to at least 30 subjects, and then your sampling distribution will approach normality. But that's not to say that your data itself will be normal with 30 data points. So this is where a lot of students get confused because you can have 30 data points and they be not normally distributed. So reaction time is a great example of this. You can have 30 different reaction times and they probably won't be normally distributed because that's the nature of reaction times. This rule of 30 is based on central limit theorem and the sampling distribution, this hypothetical distribution that we're estimating. That's also not to say that the rule of 30 is enough to get you an effect, right? So your effect size, you might need a larger, more than 30 if you have a small effect size to be able to detect an effect. The rule of 30 is just enough to meet this sort of normality assumption of the sampling distribution. The second reason to care about normality is assuming that you have your random sampling and your independent observations from a normal distribution, then your sample mean and sample variance are independent. So whenever we take the sample, we use that sample mean to estimate both the mean and the variance about the population. So you know, when we're saying, okay, we're using our sample mean to estimate the population distribution because we don't know if this sample comes from a different distribution. We're also going to be doing that about the variance once we don't know the population standard deviation. And in general, that's what you're going to encounter. So if you think most of the time, whenever you collect data from a sample and then you're generalizing it to a population, you almost never have information about that population in terms of its standard deviation, unless you're working with like census data or something like that, or some IQ data, things that have been normalized and standardized. We generally don't know the population standard deviation for, I don't know, visual cueing or visual search or whatever you're studying, you know, or the attentional blink, for example. You generally don't know what the population standard deviation is for that. So we have to estimate it. So normality is important in this sense because it ensures that the mean and the variance are independent. These estimates are independent. If it's non-normally distributed, those two errors will be related, and then you're going to overestimate the mean, and then you might over or underestimate the variance. It depends. Basically, this creates a mathematical nightmare is the take home of this. So normality, try to get normally distributed. We try to make sure your errors and residuals are normally distributed. Make sure you don't violate this assumption. So how to assess this? So we'll go into R in just a second, and I'll show you how to do this within R. But essentially, because we don't have access to our sampling distribution, right? Because we talked about how this is a hypothetical distribution, we usually test the observed data. So we can't actually test the sampling distribution because that's that distribution. If I were to, an unlimited number of times, draw samples, calculate the mean, draw samples, calculate the mean, but I don't have access to that. So we're just going to use our sample as the proxy for that. So in general, if our N in our sample is greater than 30, we're going to assume that the sampling distribution is normal anyway, because it's the general rule, and that's generally what you see with sampling distributions, even if the data itself aren't normal. So very first rule, if your N is equal to or greater than 30, we're just going to assume the sampling distribution is normal. Then we can also visually look at this. So we can plot the histogram of the actual data and a QQ plot, which is going to plot those residuals that I was talking about. And I'll show you that in just a second. And then lastly, we have a statistical test where we can actually look at statistically whether or not the skew and kurtosis of the distribution violates normality or is statistically skewed or has a statistically high significant kurtosis. So essentially what we're doing here is we're quantifying the skew and kurtosis of a distribution. So if you think back to like the third or fourth lecture when we talked about skew and kurtosis, remember leptokurtic, platykurtic, all of that. So you can actually quantify these values. So we're going to quantify those, convert those to a z-score, and then you just, it's basically a statistical test. Is that z-score significantly higher than a cutoff value? So, and I'll go through this in R in just a second. Also, there are other formal tests called, I'm not going to try to pronounce this, Kolmogorov, Smirnov, Shapiro-Wilk, Anderson, Darling. So there are a bunch of different tests here. Usually I use the Shapiro-Wilk, but about a lot of different ways to do this, which again, we'll go through. I think the only one I show you in R is the Shapiro-Wilk actually. And these are actually quite easy because this is where we're going to use some R packages to help us. We're just going to install some R packages, And there are already built-in functions to test this stuff for you. All right, so I'm going to go into R. If you want to also hop in there, there is some, I think I already have some code written in there where we can test some normality. All right, so if you come down to, for me, it's line 67, might be a different line for you guys, but it's essentially after we did our z-test here. So the very first thing we're going to look at is this, some of the built-in data. So base R also comes with some built-in data. The MT-CARS data set is one of these data sets. So if you're just already in R, if you just call this data set MT-CARS, it's MT, not empty, CARS. This is already a data set that exists inside of R. So we're just going to use this sort of fake data here. And you can see it has different cars, miles per gallon, cylinders, horsepower, all this stuff. So let's say I want to plot a histogram of miles per hour. So you can see here, it's very much not normally distributed, right? There we go. So do you see how we can see it's kind of normally distributed, but we have this weird bump thing going here. So it's more rightwardly skewed.
But you said miles per hour, but we're doing miles per gallon.
Miles per
Yeah,
gallon.
right? Yeah, okay.
Yes,
No worries.
you're right. Yeah, miles per gallon. So I guess this is fuel efficiency based here. So you can plot both the histograms and the density functions. I'm not going to go too much into this because we've already talked about these density functions and overlaying the density lines on histograms and stuff. So it's really just to show you another way to look at your sort of density function here. Okay, so I'm actually going to skip down here to add an outlier. So you can see what the data looks like right here. If I'm going to create another column called miles per gallon 2, so I don't overwrite the current one. And so I'm just going to assign that to the original column. And then I know the length of MT-CARS. I know the length of this is 32. So I'm going to change that last data point. Right now it's 21, but I'm going to make it more extreme so we can pull the data out so you can see what the distribution looks like. Also, these are not, you would never be changing your data. These are all for illustration purposes. So now I have this miles per gallon 2 where I've changed the last observation to 50. So let's say we added in a Tesla or something, or Prius or some, I don't know, some kind of hybrid car in here that has a high miles per gallon. All right, so now if we did that same plot, now do you see how this one is way out here? So this is our outlier all the way out here. And if we plot our distribution, you can see how the tail is being pulled all the way out. So now,
Wait, okay. I might have missed, I'm trying to follow, but mine's looking kind of different. You just ran 107 to 112?
yes, so if you run from 100 to 116, if you run that, you should get something like this.
Okay, thank you.
I kind of missed the change you made after the length. Can I quickly look at that?
Yep. So this, I'm just changing the last value. It's this right here. I'll pop that in the chat. So this is not really important in terms of normality or anything. This is just to illustrate a point to show like we have this skewed distribution, or I created a skewed distribution, right? Just so you can see, you can compare what happened. So we went from a relatively normal distribution here. So we have a few bumps, but again, this is what real world data. So, you know, we always plot these very pretty bell curves. That's not what your data is actually going to look like. It almost never will, because that's not how real world data is. It's messy. So something like this is relatively normally distributed, but imagine if you had that one outlier, let's say that one reaction time that that person was waiting, you know, a second and a half to respond. You're going to see distributions being pulled out like this, where there's one data point way out on the end. this is also this is why it's good to plot your data. Let's say if we had initially, if this is our original data and we plotted that and we saw that one bump there, we would be like, oh, that's weird. We didn't have any hybrid cars, and I can't think off the top of my head any regular cars that get that high miles per gallon or something, right? It might be like a red flag that something's wrong with your data. So in general, this is just a PSA on plotting your data histograms whenever you first get it, just to sort of see what your distribution looks like. So now if we want to hop to looking at actually plotting out normality and testing it both graphically and statistically, let's load in the festival data. So this is a .dat. So we're going to do read delim. Oh, no, we're not, apparently. Not in the right. So make sure you set your working directory. There we go. And see this header statement again, just looking at the first few rows. So we have our gender, day 1, day 2, day 3, some kind of festival data. Oh, you have the ticket number. There we go. So now if we want to plot a histogram, just like we did before, let's say we want to plot a histogram of just day 1. So you can see here. So do you see that one way out there on the edge at 20 something? So you can see there's an outlier. Does everyone see that?
Sorry, I'm still a little delayed. Okay.
Got it? Does anyone else having issues getting the data read in or anything? So these are just, we're not going to spend time making them very pretty. This is just for graphical purposes. Oh, if I wanted to add some numeric. So, you know, we talked about plotting those density lines as well. Oh, apparently this is not going to work. But it's fine. We'll just do the histogram here. This is enough to see that we have this outlier going on here. So one thing we're going to do is say, okay, we're going to remove this. We'll do DLF day 1. And there are also a whole host of ways that you can do outlier removal. So usually you don't want to just eyeball it like this. You want to like standardize and say, okay, anything that is more than like 2.5 standard deviations away from the mean, then we're going to remove those outliers. You have to have some sort of criteria, but just
Yeah, because just from looking at the histogram, you can barely see the outlier. So you wouldn't suggest looking at a histogram and deciding what the outlier is, right? Like you would have to run some statistical methods.
Yeah, so usually this varies from lab to lab how you determine it. So sometimes it might be something that's actually statistically like so many standard deviations away from the mean, or it could be something that's implausible. So for example, if you have a task a cognitive task that you know on average takes people that they cannot do it any faster than 300 milliseconds, and you have someone who's starting with 100 milliseconds, even if that's not one standard deviation from the mean, that's an outlier because they're not doing the task, right? So whenever you're dealing with cognitive constructs where you're like, I know physically in the brain you can't do this quickly, so this is an outlier. With behavioral data, you can do it that way as well. But if it's something like, you know, let's say they're just responding too slowly, you might want to do like a cutoff of like 2 standard deviations away from the mean or whatever.
So in which case you would run like, look for any answers that's like less than 200 milliseconds and make a fault like true or false and find a false and call that an outlier kind of, right?
Yes. Yeah. So that's one way you could do it, or you could remove it and just replace it with
an A.
A's, yeah, where you're just like, I'm not this person, you know, if all of their data is an outlier, you might end up removing the whole subject, like if all their data is weird. But if it's just that one data point, maybe you just replace it with an NA. There are a ton of different ways to deal with missing data, but
there's no one specific way that you're suggesting right now.
No, not at all.
Okay.
Yeah, because that's going to be very specific to what you're studying, the lab you work in, and it's
what data it is,
what the data is, what you're looking at. So it's very, there's a whole host of ways to look at that. Yes.
Got
I
it.
think we have some extra slides I can probably upload to Blackboard with the different ways to deal with outliers, but there's a ton for sure. So, and as we can see here, look, our mean is 1.79. Because I can't remember what this is measuring, but our mean is 1.79. That little outlier is 20. So I know that is absolutely an outlier. So even if we said 3 times the standard deviation, which is pretty strict cutoff. So if we added 3 times the standard deviation to the mean, just to see if it is. So we could say, oops. What happened there? So really anything that is 4.62, that's greater than 3 standard deviations away from the mean. So in general, you don't always want to trim off. You don't usually want to trim off too many outliers because sometimes there's information in the outliers. You know, sometimes those are the interesting cases. So if you end up with a lot of data out there because of the way the distribution is shaped, you don't want to trim those off. But when you have this one data point that's sitting at 20, we just want to get rid of that. So here we're going to, so down here I'm going to say, okay, if, so there's a statement called if else. So this, you give it the logical that you want it to test, and then what you want it to do if that thing is true, and what you want it to do if that thing is not true. So I'm going to say if else day 1 is greater than 20. So if it's, because we can see out here it's already past 20. If it's greater than 20, I'm going to replace it with NaN. Else I'll just replace it with the value itself. Does that make sense? So now if we, so there are 800 of these values, so it's probably not going to show all of them, but now you can see if we plotted this again, that value is going to be gone. So let's grab our histogram. And now do you see, now we actually do have a normally distributed data. So do you see how that outlier was, because our histogram plot will try to automatically determine the size of the bins. You can go in and control that, but just when you're briefly looking at your data, letting our automatically choose the size of the bins is a great way to show your outliers, because it's immediately obvious that something crazy is going on here. And then when we remove it, now we have a normal distributed data. And notice how the axis changed from 0 to 4, because R was trying to automatically put all the data because we had that outlier.
So if I get error in eval, EINVIR.
Error in, say that again. In
Let me just copy and paste.
oh,
But I don't really have anything that says D. This is what I have. I don't know where the object D is coming from. Oh, yeah.
it did that again. Let's see.
Oh, well, I just ran it again and it's fine. Never mind. Sorry.
Yeah, that is weird. I'm not really sure. But it's right for me.
Mine is giving true false output instead of numbers.
Okay, so it's giving true false.
Like I'm getting a ton of falses, that like a block of text like in the console for you, but it's not numbers, it's just false and false, 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 false.
Is it like this?
Oh yeah.
So
Oh, so I need to,
so you have
but
to
that's
run the whole thing. Yeah. Let's-
okay. Yeah, I did run that line 130.
Oh, you did.
Yeah, and that's what I got from it.
That is
Is
interesting.
there something else I have to run?
No, it should just be this line there. I'll paste it in just in case there's a weird parentheses or comment issue. But yeah, so because we've already assigned it to a variable, it shouldn't actually print anything to your console. It should just do it should be nothing down there. I only have it running because then I highlighted this section to make it print.
Oh, I forgot the arrow. Wait, hold on.
Oh, to assign it. Yeah.
Yeah, I like did a greater sign.
Yeah.
And then you,
Okay.
sorry.
All right, thank
And
you.
then And the new histogram is supposed to be from 1 to 4, you said, right? Like the one that I pasted.
So now we can see our histogram for day 1. But remember, we actually have day 1, day 2, and day 3. So I'm going to plot all 3 of these. So if what we were interested in is testing, the differences between day 1, day 2, or day 3, or whatever our hypothesis was, generally we're going to want to test the normality of those distributions separately. Because think about this, if our hypothesis, if our null is that they're the same, but what we're actually testing is that there's a difference between day 1 and day 2, then whenever we're looking for normality, we shouldn't collapse those together, because we're already thinking that there's a possibility they're different distributions. So we need to look at their normality separately. Does that make sense? So if that's in fact what we're testing, then we should be also looking at their distributions separately as well. And as you can see, this one is distributed differently. This one is rightward skewed. And then we have more of a rightward skew here, but a little bit more bumpy here on this one. So any questions about this? Looking at distributions, sort of assessing, pulling outliers. This is just general, like scanning through your data, making sure there's nothing crazy going on, looking at the different distributions. So now I'm going to run this NA omit. So this is if there are any NAs in there, which we know there's at least one, right? Because we put that one in there. So I'm going to run this. So that will delete that entire observation. So now notice, you see how we have 809? If you look over in your global environment here. But remember up here we had 810. So all we did was just remove the person that was an outlier. All right, and now I'm going to.
Is that, sorry, how's that different from what you did earlier about assigning it through if else?
So this gave it a value of NaN. I don't want it in the data at all.
Okay.
Okay. Change the data itself. So that's why I'm creating this other vector, but I just don't want it there at all. So we can plot, because some functions in R have problems with dealing with NAs and NaNs.
Right, because when we plotted the new day 1, it actually shrunk the size. Well, the histogram was smaller because it's not counting the outlier or the now NaN. So I wasn't sure what's the difference of making it NaN where the histogram doesn't even count that as a data versus omitting the data.
So because you're right, the histogram will ignore it, but the thing that won't ignore it is this density function. So
okay.
So whenever I want to plot, if you wanted to plot the line over top of it, so this density line to have the nice distribution line, it won't do this density function with an NaN in it. So
what would it look like if not?
So we can actually do this up here. So I'm just going to scroll this up here to add it to this. I'll just move this to here.
And that's just with an NaN, right? Yeah.
Yeah. And so do you see this error? X contains missing values. So because it has an NaN in it, so it doesn't like it.
So it doesn't even do the density line.
Yeah, so it won't do this calculation. So if I just ran this one part, this is the error I get. So it won't do anything with it. It'll just throw an error.
So for example, if you didn't put an NaN and kept the outlier, then it would just bring you a different density line. If you make an NaN, it doesn't give you a density line. And if you omit it, then it'll give you the more correct density line.
Exactly. So if I'll just load in the data again and not deal with the outlier. So this, and now it'll give me the density line, but do you see how low it is?
Oh, yeah.
It's like all sorts of messed up, right? So
okay.
Because it's
not even correct density line. Yeah.
Yeah, because it's being pulled all the way out there. And also because this is, well, here's one other problem with it. Hang on. So whenever you're plotting density lines over a histogram, you have to do this frequency equals fault. So do you notice the axis here says frequency? This is one reason why it's flat. There you go. So frequency
Okay.
false changes this axis to density, but do you see how it pulls the density line all the way? And then there's that little blip right
Yeah.
there. That's, we know
Right.
that's like bogus data, right? So we're just gonna get rid of that.
Right. Okay, so when you didn't set the, whatever it was to false, what did you call it? The frequency
Frequency.
equals to false, then it just gave you just an incorrect density line. And when you set it to false, it gave you a correct one for the data that's given that includes the outlier. Okay.
It will give you
Alright.
a correct density line. It's just giving you the wrong scale. So notice the scale of
Right.
density is 0.0 to 0.3.
Yeah.
And the frequency, if I change this back to true, look at this
It
scale.
just gives you like a weird flat. Yeah.
Because the density line is correct, but it's so tiny that you can't see it because it's trying to
Okay.
plot 3/10
Okay.
point down there.
That totally makes sense. Thank you.
Okay, so we have our NANs being removed, and then we have our density line. So we have a pretty normal distribution here. So now we can do the exact same thing. I'm just gonna copy and paste this for day 2. The only thing I'm changing here is the 2. I don't think that we have any NANs in day 2, but I'm just gonna go ahead and remove them anyway. So if there are no NANs, this won't do anything. So it's fine to do either way. It'll just make sure I don't get any errors. And now you can see for day 2, and then we could do the same thing for day 3. So this is not anything too crazy, just looking at, again, our distributions. So of the 3, day 1, day 2, or day 3, which one appears to be normally distributed?
Day 1.
Day 1.
Day 1, exactly. And but that's, remember, only after we remove the crazy outlier. So, but sometimes that is something that you have to do to deal with your data. If you have something that is not in line with, something that you should expect, something like a very slow reaction time or a way too fast reaction time, or something that's that crazy, that's okay to remove, as long as you have some sort of standard that you're applying uniformly. And then now you don't have a normality problem with this one. And you can see these are rightwardly skewed, but because we have 810 observations, we've already met our N of 30 for central limit theorem. So we don't really have to worry about our sampling distribution normality. The only thing now we have to worry about are the residuals. And so this is where those QQ plots come in. So the way that you plot a QQ plot is either QQ norm or QQ line, depending on how you want it to look. So QQ norm is going to plot basically all the sample quantiles by the theoretical quantiles of what they should be if they are normally distributed around the mean. So this is basically should be a straight line is what you're looking for. And QQ line will do this as a, whoops, I see what I've done here. Disregard that because I plotted 2 different ones on the same line. There we go. So I don't know if you can see that line that's under there. So these are so normally distributed that you can almost barely even see the line. We'll see a difference here when we get to Q2 and Q3. You can also scale the data whenever you plot it. So this puts it on a standardized scale where it's scaled relative to the center or the mean. So everything is how we were talking about the residuals being distributed around the mean. So now they're just all on the same scale. But you can see they're basically giving you the same piece of information. The only thing that's changing here is the scale here, whether the 0 is the mean, right? So this is, think of this when we did our Z transformation, the mean is 0 and everything is less than or greater than the mean. It's the same thing here when you scale it. So it gives you the exact same plot. So usually I just do it with the raw data, but you can do it either way. So I'm going to copy and paste these down for day 2. Make sure you just change these to 2. So again, this is the way that we're graphically looking at normality. So now do you see how this one is all kinds of crazy, how it's shifted up a bit? It's actually not that bad, but you can see where the line is here, and this is not following that line so well. So this is actually probably a time where scaling it is a little bit better, but there we go. So you can see how this is a little crazy. It's deviating quite a bit, so it's not great. It's not the worst, not as good as day 1. Ideally, you want to see it like you see on the day one data.
So what's the difference between the scaled and the not scaled ones?
The scale just changes the axis here, the sample quantile axis to be relative to the mean. So that the center is 0. So if you see this, so it makes it relative to the mean versus the raw data. So it gives you the exact same plot. It's just changing the scaling on the side to make everything relative to the center point.
Okay.
I say the mean, but it's actually the center point. All right, so let's look at 3. And so 3 is our worst one by far. So do you see how these are really deviating from our line here? So this one is definitely not very normal. So whenever you're looking at these, you definitely want to see something like this. That tells you that your residuals are relatively normally distributed. But again, this is all subjective because you're just graphically looking at it, right? So if you have something like this, where you're like, oh, I don't really know. I mean, this one's actually pretty bad, but if you're like, oh, I don't really know, then we can do a statistical test of whether or not you violated normality, because this is just a graphical display. We can actually test it using those skew and kurtosis values. Was there a question?
Oh, yeah, Ali, the day 3 is worse than day 2, even though it sort of seems like it balances with some below the line and some above the line, where everything on day 2 that was off was above the line, but it's just more wacky. So the day 3 is worse, huh?
Yeah, I mean, I guess day 2 is actually, now that you said that, day 2 is actually pretty bad too. Yeah, because you do want them relatively close to the line, both above and below. So, but you do have a lot here that are kind of right on the line. So I don't know, it's graphically, it's hard to say which one is worse or better, at least for me, because I feel like it's so subjective. I can just tell you if it's good or not good. So then if I have these like questions, that's when I defer to like, okay, let's look at this statistically and see like, does this violate normality?
But it doesn't balance it more, even if they're way out, to have some below and some above the line, as opposed to everything above the line. That's just what I was wondering about.
Yeah, so ideally you just want to see them hugging that line all the way, because these theoretical quantiles, they should be creating a diagonal with the theoretical quantiles.
I had a quick question.
Go ahead.
So like when the points kind of like adhere to the line, that just means that the residuals are more normally distributed as opposed to if they were like off of the line?
Yes, yes, because the line is telling us how we would expect them to fall if they were normally distributed.
Okay, cool. I just wanted to make sure. Thank you.
All right. And that's why, because we're applying them against the theoretical is what we would expect. And then we have our actual, so we would expect it to be a diagonal line.
So does that mean the points which are not on the line are far away from the standard deviation, kind of like?
They would be far away from, well, they're not necessarily far away from, it's not necessarily that it's just far away from the mean, but it's that it's unbalanced from the mean. So it's not, the residuals are, you know, we talked about like if your mean is 10 and one data point is 7 and one is 12, then that means you have a -3 and a +2, right? So you want to see your residuals being balanced around the mean or distributed around the mean.
Okay.
So basically you want to see it hug the line. That's the go-to I tell students. And then if you're unclear, which is a lot of times what I do, because I hate when things are subjective like this anyway. So we want to test it statistically. And a lot of times if you just see something that's pretty close, I mean, like this is like very, very good distributed data. But if you see something that's even close to this, then you can probably just say, yeah, it's good and move on. But if you really want to test it, then you can use some of these packages. So if you don't already have this, you want to, in the site package, there was probably something when you opened R, a little yellow window up at the top that said that you need to install some libraries. If you didn't click on that button, you want to do install.packages and psych. I'm not going to run this because it will make me restart R because I already have this installed and it will try to update it. But if you
If
don't
it
have,
shows, sorry, if
ahead.
it shows up as a library psych and the blue thing in the bottom when you run it, it means you have it, right?
yes, so if you do, yeah, so if you try to tab over for psych, if it shows up down here, then that means you've already installed it.
Cool, thank you.
And also you can check for packages over here. So they're all the ones that you have loaded are these. So you can see, actually check mark them and search them and everything. Okay, so I'm gonna use the psych library here. So one helpful function is the describe function. So this gives from the psych library. So this only works after you load the psych library. This gives you the number of observations, the mean, standard deviation, median, all of that, the minimum, maximum range. But what we're interested in is skew and kurtosis and standard error. So it actually already spits all this out for you. So this is also helpful in other regards, but it gives us these skew and kurtosis values. Notice the skew is 0. Why does this make sense? Let's go back to our plot. This is the one that we're looking at. Why would skew be 0?
Because it's pretty normalized.
And it's close to the mean.
It's symmetrical, right? So skew is whether or not the tail is going right or left, but this one's pretty symmetrical, so it's 0. If we had not trimmed that outlier, that would not be the case, right? It would be have a positive skew. So you can also do, oh, go ahead.
It averages because skew 0 seems pretty hardcore.
Sorry, say that again.
Was it just a skew of 0 seems pretty hardcore? Like what does it averages? Like how does it like what are the ranges of skewness that it?
I don't know just from this package. I think it just calculates the skew. It's probably just rounding though, because it looks like it only goes out like 2 decimal places. So it might not be perfectly 0, but it's probably just rounding it to 0 here. And so you can also do all 3 at once. So day 1, day 2, day 3 here. And now you can see we have skew, skew, and skew, and our kurtosis for all of these. So we can actually look at these skew and kurtosis values ourselves. But really what we want to do is find a way to statistically test whether or not they are statistically or significantly skewed or have a significant kurtosis and away from the normal distribution. So I'm going to come back to the way we can do this with the describe function, but first there's another function that'll just do this for us. That's the way I err, because I think it's better to be efficient. So the pastex library here, if you haven't installed this, you'll have to do that, just like we did the psych library. The function you want is called stat.describe. So I'm going to load that and then stat describe. And now you'll see down here, this has this kurtosis 2SE, and there's also a skew 2SE. Essentially, the criteria that you're going to adhere to is if this value is more extreme than 1, can be positive 1 or negative 1, because remember, we're dealing with distributions, both two-tailed situation. If it's more extreme than 1, then it is significantly skewed. And so usually we would say something that is more than, because these have been converted to z-scores essentially. So, or this has been converted to 2 times the z-score really. So usually
they're more extreme than positive or negative 1, correct?
Yes, positive or negative.
Okay.
And usually we would say, well, 1.96 is our standard cutoff for a z-score, right? For an alpha of 0.05, a z-score is 1.96. So usually we would say, let's round that and say 2. But this is why this is the 2 times the standard error. Just, it already does that math for you, basically. So we could solve this algebraically. So if I took, for example, I'm just going to take this from day, looks like it's taking it from day 1. Let's take this from day 1. This X variable, this just turns this into a data frame. So I just turned all those values into a data frame. And then if I grab the skewness and divided by the standard error, which remember, that's how we would convert something to a z-score, right? grab your value divided by some error term to your like Z observed. And notice I'm multiplying by 2 here because this is 2 times the standard error. So I have to get it back up to where it's supposed to be. So this is the actual standard error. And then if I converted to a Z-score, this is skewness divided by standard error. So this Z-score, Z-score here will equal, this should equal 2, hang on. Oh yeah, divide this by 2. So the Z-score divided by 2 equals the 2 SE skew that it gives out. So basically, if we were converting this to a Z-score, we would compare it to 1.96. So this Z-score, this isn't a good one because it doesn't violate. I mean, I guess it's a good thing, but this Z-score is negative 0.036. What the skew to SE gives you is half of that. So since we've half that, we half our criteria. Usually our criteria is 1.96. Let's round up to 2 for simplicity's sake. Half of that is 1. So if this value is more extreme than 1, you say it's statistically skewed. Same thing for kurtosis.
So my answer is coming up as 2.3, and I have no idea where the number's coming from.
Oh, change up on line 196.
Yeah,
Is that day 3 or day 1?
day 1.
Oh, that is? Okay. Interesting. So let me paste this in the chat. Let's see if you guys get the same thing. So
Yeah,
essentially, is it still coming up that way?
let me, I didn't change anything on the original thing that was in there. Oh, nope. Yeah, okay. No, it's coming up.
Yeah, mine
Uh,
was day 3.
Yeah, mine was right here, day 3.
Oh, the bottom, okay.
Yeah.
Okay,
Yeah.
the stat dot dissect was day 1, but the as data frame was day 3. My bad. Yeah.
I think that was a typo. So yeah, that was typo on my part.
No,
Yeah.
no.
So yeah, so that should give you, so essentially this is just showing you algebraically why this math works out. You guys don't have to go through this step all the time. All you have to do is this stat describe and look at the skew.2SE and say, is this more extreme than positive or negative 1? If yes, it's significantly skewed. If no, you didn't violate. So this is when you're testing assumptions, when we do statistical tests of assumptions, this is where some students sometimes get confused because usually we think of having a statistical result, statistical significance is like a good thing, right? But if you're testing assumptions and it's statistically significant, that means you violated that assumption because that means that thing that you're testing, skewness, for example, is statistically different than normal, and we're testing for normality. Right. So if it is different, you violated the assumption. So here we have not violated the assumption. That's good. Here we have. So do you see how this is greater than 1? And we could also test this the exact same way. So I'll show you down here for kurtosis. Except here, I'm just going to change this to kurtosis, and this should just be kurtosis to SE. And then we'll see that we'll get the exact same value here. Oops. So let's say first we can get the standard error because this is 2 times the standard error. So we need to get back to standard error. And then Z-score. So our Z-score here for kurtosis is actually negative 2.46. If this was a standard Z-test, we would be comparing it to 1.96, right? So do you reject the null or fail to reject the null?
Reject the null.
So we reject the null, correct. That is that we have significant kurtosis. And that tells us the exact same thing. If we divided that number by 2, that's the exact number. Oh no, what's happened here? X kurtosis. This should be. It should be the same number. Oh, I see the problem here. Actually need 2 SE. There we go. Yeah, so these numbers are the same. So dividing that z-score by 2 is just the same thing as this. So because we're halving the z-score, we just half the criteria. So instead of comparing it to 1.96, we're just going to compare it to 1. And I know that 1 is not exactly half of 1.96, but for all purposes, we're just going to round up to 2 and half it, just for simplicity of math. So go ahead.
I'm not sure why we're halving it though.
So the program, the stat describe function itself does that. So that's just the value that it gives out to make it easier to look at. So you can just quickly look at this, and all you have to say is you look at this CURT2 SE, and if it's greater than 1 or more extreme than positive or negative 1, it's significant. You also can look at this one, more extreme than negative positive 1, just look at that one. It's just for an ease of looking at it and using 1 instead of 1.96.
Right now it's violating it, right? Based on
Exactly,
the
for
state.
kurtosis, yeah. So we're violating kurtosis, but not skew. So, and almost always with real data, you'll find that you probably violate one of these as long as you're not violating kurtosis and skew, and it's non-normally distributed, and your errors are all messed up. Like if you're violating every single thing, then you might need to look into some remedies for normality. But given how normally distributed this one already looks, I would probably just say it violated kurtosis and then just move on with it, because it's not that big of a deal. Skew is way more problematic, right? Because when the tails are pulled out on one side or the other, that changes those probabilities. Well, technically it doesn't, as long as your N is greater than 30, because your sampling distribution is going to be normal regardless.
So violations of kurtosis in some way means that your data may not be normally distributed,
It means
or
the raw data is not
Uh-huh.
exact. It's not perfectly normally distributed, but again, that's not, if you have an N of at least 30, then you don't have to worry about it for like central limit theorem assumptions wise. The only thing you have to worry about is for like your estimates, making sure that they're independent of one another, or how the residuals, sorry, that was the other thing. So as long as your residuals are normally distributed, then that also is not really that big of a thing.
okay.
All right. So basically this is the rule. If it's greater than 1, it's significant. And here, if it's significant, that means that you violated the either skew or kurtosis. And then the last thing that we'll do is the Shapiro-Wilk test is an actual test of a formal statistical test of normality in general. So, so this is called the Shapiro-Wilk normality test, and this is going to automatically spit out a p-value. So even here you can see, as normal as this looks, technically this still does violate normality. So it's like p of 0.03. So yeah, again, most people aren't going to these great lengths to test all these things. They're looking at the residuals. If they look pretty good, they're rolling with it. But technically this is the statistically appropriate way to look at all these things. So, and again, if you have, I think it's less of a problem if your sample size is at least 30, because the more important thing is that the core assumption of a lot of these tests are based on that sampling distribution, right? The normality of that. So if you have at least 30 observations, your sampling distribution is going to be normal. So this isn't as big of a problem. But if you find it to be problematic, you can always look into, other data cleaning. Maybe you need to clean out some more outliers or have some sort of like criteria, something like that. Yeah, so the Shapiro-Wilk, this is just Shapiro.test, and then you just pass the variable that you're wanting to test here. So super easy to run, and it spits out a p-value right for you. So you don't have to do any extra math or anything like that. Just kind of nice. So are there any questions about that? All right, so I'm gonna hop in. So we have about 15 minutes. I think we can get to some of these t-tests. So and all of this information is in these PowerPoints also, so you can go back and look at all that. I'm not going to talk about homogeneity of variance yet until we've already covered the t-test, because you don't really have anything to anchor that to, because we haven't talked about separate groups yet. So I'll come back to that. That assumption doesn't apply to z-test, one sample z's, because you only have one sample. So once we get to 2 groups, I'll come back to homogeneity of variance. All right, so one sample z-test we just talked about, right? So you have, let's go back to our IQ example. Mean of 100, standard deviation of 15. We wanted to compare students with a mean of 103, right? But let's say that we didn't actually know the standard deviation of the population, which is often the case. We rarely actually have access to that information. This is when this occurs, you can no longer use a one sample z-test, and you have to use a one sample t-test. And so we have to rely on, oops, sorry, my PowerPoint is kind of wonky there. There we go. So you have to rely on a t distribution instead of a z distribution. So remember, up until this point, we've been looking at the z distribution, or the standard normal distribution, right? And we've been using p norm, q norm, all of that. And everything's been in z-scores or standard deviation units. But often we don't know the standard deviation of the population. And when we don't know that, we can't use the standard normal distribution, because that's based on knowing the population standard deviation. So if we don't know that, we have to default. If we don't know that, then let's think about what we use the standard deviation of the population for. When you do a one sample z-test, why do you need to know the population standard deviation? Where do you use that information at?
Is it calculating the standard error?
Exactly. When we calculate standard error, because we're trying to get the standard deviation of the sampling distribution, right? Because we know standard error is the standard deviation of the sampling distribution. And we know that is going to be related to the population standard deviation and your sample size. So remember, with central limit theorem, the larger your sample size, the skinnier your distribution is, the smaller your sample size, the fatter your distribution, which is going to relate to the standard deviation. So larger standard deviations give you fatter distributions. So we need that population standard deviation to calculate the standard error, AKA the sampling distribution standard deviation. But if we don't have the population standard deviation, how do we calculate standard error? So this becomes a problem. And we often don't have the population standard deviation. So what we do instead is what we love to do in statistics, is estimate something. So we love to estimate everything, right? So we're estimating the population with the sample all the time. We'll just do that again. So we can estimate the population standard deviation using our sample standard deviation, and then just calculate standard error that way. So now notice our new standard error formula. Instead of sigma divided by square root of n, it's now s divided by square root of n. So remember the differences in symbols. S stands for the sample's standard deviation, not the population standard deviation. So now when we calculate the test statistic, because we are no longer using the population standard deviation, we're using a surrogate for it, because we're estimating what it is based on the sample. We no longer can use this standard normal distribution. Now things are going to be shifted onto what we call a t distribution, or it's also called the student's t distribution, developed by a grad student years and years ago. I think he worked for Guinness, actually. He was like trying to map out things for estimating, I think like the ABV and the beers or something a long time ago. There's a whole history thing on it. I'm sure you can look it up. Okay, so students' t distribution. So because we have to estimate the population standard deviation, now we use a one sample t-test instead of a one sample z-test. Notice the similarities in these formulas. What is the only difference?
S versus sigma.
Yes, S versus sigma, because here we're estimating the population standard deviation using the sample standard deviation and therefore calculating standard error. And here we have the population standard deviation. So the reason that it has to shift to a student's t-distribution is now that we're estimating a population parameter, we now have to consider consequences of this. And this is another place where our thing that we've learned about before, degrees of freedom come into account. So anytime you make an estimate, now we're going to lose a degree of freedom. So we've estimated the standard deviation of the population, lost a degree of freedom here. So just keep that in mind in a second. So now we have to also consider that we're adding additional error, right? Because we're using our sample's standard deviation, which might also have error in it, to estimate the population standard deviation. So because there's going to be more error, we might be overestimating, we might be underestimating, and we have to account for that. And that's what the student's t-distribution does, is it basically penalizes you a little bit for having to estimate this. So it's going to make it slightly harder to get a significant result than on a z distribution, but it should be, because we're adding error. The more error you have, the more you want to make it, you want to penalize yourself a little bit so that you're not committing type 1 errors left and right. So if we look at our sort of example here of z distribution versus a t distribution, you can see here how the t is slightly skewed here off to the right. And because this allows us to use our S squared or variance, remember this is the symbol for variance, as an unbiased estimator of our population variance. So the sampling distribution of our sample of standard deviation or sample variance here, if you see here how it's slightly pulled out to the right, so it's slightly skewed. And you can see here how the population standard deviation variance, I know this is probably a very confusing chart because this is the distribution of the sample variance and it's the sampling distribution of that. So imagine, remember when we created the sampling distribution by sampling, calculating a mean, plotting it, sampling, calculating a mean, plotting it over and over again. Imagine if we did that with variance. So if we did it over and over, but instead of calculating the mean, now we're calculating variance from the sample. So exact same thing, it's still a sampling distribution, but now it's of the sample variance. So you can see if a sample size is 5, again, we're getting a skew with a sample size of 30. Again, back to central limit theorem, it's approaching normality. So when you have small sample sizes and you're estimating, then you are more likely to make an error, which again is not surprising. You're more likely to overestimate, right? So this is not as surprising because we know based on central limit theorem, as n goes up, standard error goes down, right? Those two things are interchangeable. Does that sort of make sense? This is all the same basis as a sampling distribution with means, but we're just using variance now to illustrate the same concept. So because of this, we tend to, oh, I'm sorry, I said overestimate, I meant underestimate our population standard deviation, particularly for small sample sizes. And remember, if we're underestimating, we're saying that there's less error than there actually is. So we have a small sample size. We already know there's a lot of error. And now we're saying, oh, I think that there's actually less error than we're underestimating how much error there is. So this creates a problem in that it's going to make it really easy to reject the null, potentially creating a type 1 error situation. So the student's t-distribution is essentially just a way to correct for that and take into account the sample size. And the sample size will be reflected in your degrees of freedom, which is going to be, does anyone remember what our formula is for degrees of freedom? We've only estimated one thing, so I'll give you that much.
n-1.
N-1, yes. So degrees of freedom here is going to be n-1 because we've estimated population standard deviation. So we're going to lose that 1 degree of freedom. All right, so we're going to use our sampling distribution, but now our t distribution instead of our z distribution. And whenever we're calculating our sample variance, remember, we're going to divide by n-1 because we're using this estimate. So we're losing a degree of freedom instead of dividing by n. And then we're going to be able to calculate a t-score instead of a z-score. So all of this is very similar to what we did with the one sample z, but now we're doing it with a one sample t. Does that make sense? So basically, this formula is going to be identical to what we've done with a z-score. The only difference is instead of you calculating standard error with the population standard deviation, you're going to do it with the sample standard deviation. Make sense? So we're almost out of time, so I don't think we have time to jump into the R bit yet, but let me just go through this. We'll come back to that next week. And let me just go very briefly through these differences, because once you sort of see, as we go through the course, all these different statistical tests, it's very easy to get overwhelmed with how many of them there are. But if you think of them in a pattern, think of how they're similar and where, because most of them have this core structure that we're taking some kind of mean difference and dividing by some kind of standard error. Every single test that we're going to do is basically has that structure. It's just the circumstances change and depending on which one. Here is the first chart for that. The z-test, you know sample mean, or you know population mean, you know population standard deviation. Then if it's a one sample z-test. If you only know the population mean and not the population standard deviation, you shift over into a one sample t-test. And then next week, what we'll talk about is, let's say you no longer want to compare things to the population. You actually want to compare two groups. Now you're going to shift into an independent samples t-test because you have two independent samples. Then let's say instead of two independent samples, let's say you really need a post-test pre-test situation, but remember that independence assumption we have? So you can't violate that. So then if you need to have a pre-test post-test where each person is tested twice, then you shift into a repeated measures or paired samples t-test. And so those are essentially our three different t-tests where we have one sample, you're comparing to a population. Independent samples, you're just comparing two different groups. And then paired samples, you're comparing two different or two same measures. So you're comparing two measures calculated on the same people. So a pre-test versus a post-test, or a pre-drug versus post-drug, pre-treatment versus post-treatment. Does that make sense? So just to sort of give you like a preview of the sort of different tests that are coming along. Next week we'll, so now we're almost out of time. So next week we will